And please feel free to speak in English, in Portuguese, in Spanish. We are here to help everyone to communicate in any language, even in French, if you need. So, this is the second edition of Life Cycles and DOHaD. Please be everyone very welcome. We have now 4 coordinators, Professor Fabíola Isabel Suano de Souza, from Universidade Federal de São Paulo, UNIFESP, Brazil, me, from Universidade Federal do Rio Grande do Sul, UFRGS, eu sou a professora Cristiane Matté, Professor Dr. Erika Chavira-Suárez, from Universidad Nacional Autónoma de México, UNAM, de México, y la profesora Paola Casanello, de la Pontificia Universidad Católica de Chile, PUC Chile, justamente el sitio donde estoy ahora. Estoy aquí muy cerca de Santiago, in Rancagua de Chile. So, I'd like to share with you some statistics, because we are really proud to have now 11 professionals here and 106 postgraduate students from master and PhD students. As you can see here, they are from Brazil, about 80%, from Mexico, about 17%, then Argentina, Chile, Cuba, Colombia, and also USA. So we are really, really, really happy to have all of you here from so many countries in Latin America and also USA. We have much more women, in fact, 60% from Brazil, and then Mexico, Argentina, Chile. In all these countries, we have more women than men. So the program will start today. It ends on September 12th. And we in all the days, we start 3 p.m. in Brazil. It's so if you have any question about the place in your country, please contact us. We can send you more information, but we start 3 p.m. in Brazil. We hope to end till 6 p.m. in Brazil. Today we start with some opening, welcome, and general guidelines, and then we have the theme of advocacy and DOHaD. We'll present the coordinators and the lectures after, but today we have as coordinator Dr. Elena Zambrano and Dr. Antônio Marcos Paes. Our opening lecture is, we have the pleasure to have here the president of the International DOHaD Society, Lucilla Poston. And then we have a question and answer time, like 15, 20 minutes, a little break of 10 minutes, and then a cultural lecture with Dan Saladino, and to finish the day, the question and answers. The assessment for the postgraduate students will consist of 30% participation in discussions, the presence in class, and also the participation in question and answers, and 70% of a formulary to be completed every week based in the suggested paper in classroom. So all of you probably already assessed the classroom. If you didn't access it yet, we have a QR code here you can access, or look to your email account. You probably will find there more information too. We send in the last week, I think 2 or 3 times, all the information. And the course score will be A, B, C as approved scores, and D or MRF as failing scores. The score will be determined by your participation, and the formulary will send in the classroom every week. So now you have there a paper, or in fact it's a book chapter from Lucilla Poston. You have to read, to evaluate, to discuss, and then fill the formula and send to us to have your participation in this first week. We would like to invite you to access our new website, dohadsociety.com. We have here also our big congress. We are working a lot, and today we have here one of the presidents of the congress, Dr. Marcos Paes. It will be in Buenos Aires, Argentina, in September next year. So stay tuned for more information. We will share during the course too. Here we have some of our social media, Instagram, Twitter, YouTube, also our email and our new website. So you can access it here too. And if you have any doubt about the discipline, please tell us now, send us an email after. We are ready to start if you don't have any question, but if you have questions, we are here. So, tem alguma dúvida? Tem alguma dúvida de como vai funcionar a disciplina, o curso? ¿Todo bien? Bueno. Entonces, yo invito ahora a Erika Chavira o Fabíola. ¿Quieres hablar algo más del curso ahora, en este momento? ¿Cómo estás?
Oh, it's perfect. I just would like to say thank you so much for everybody. I am very, very, very happy to stay here in the second year of our course. It's a big, very important day, or not just to connect the information, but know the people about everywhere that you are studying DOHaD concept, improve the knowledge. And today is a very special day because that will be a pleasure to me stay here with Professor Lucilla Poston. Lucilla Poston is a very, very important person that connect people around the world. Cristiane, Matté, my friend, Erika, Chavira of Mexico, Paola, Casanello, we are working together during the two or 3 months to organize this meeting. And I am very, very happy to stay here. Just to talk that we can talk in Portuguese, in English, and Spanish. We will connect in different languages because the knowledge is a universal form. Muito obrigada por estarem aqui hoje. Muito obrigada, especialmente aos pós-graduandos. I am responsible for the Portuguese language here. Muito obrigada para os pós-graduandos que estão conosco hoje, pediatras, obstetras, psicólogos, fisioterapeutas, pesquisa básica, para conhecer melhor, not just the concepts, but the leaders, the heads of the concept of DOHaD. Is a special day for me to start with everybody here. Thank you so much for this. Erika, it's for you.
Thank you very much. Beautiful words. Also, I'm very, very excited. Estoy muy agradecida, muy contenta por tenerlos a todos, a tanta gente que trabaja en ciencias de la salud. I'm very fortunate to work with you and to have here students and professionals of all health and sciences and of health. So thank you very much. I have the privilege to introduce 2 wonderful people and great researchers. Here is Dr. Elena Zambrano and Dr. Marcus Paes. First of all, I want to introduce Elena. Only a few words. She is a medical science researcher in the Instituto Nacional de Ciencias Médicas y Nutrición Salvador Subirán. And also she works in the Faculty of Chemistry in the National University of Mexico City. She got a postdoctoral training in Laboratory for Pregnancy and Newborn Research College of Veterinary Medicine, Cornell University, Ithaca in New York, United States. And currently she dedicates to development of the regions of health and disease. She is one of the first to do research of programming and aging in Mexico. She brings us this concept, so she is a pioneer here in Mexico. He has published over 114 peer-reviewed research and led more than 14 theses of undergraduate and graduate students. So thank you very much, Elena, to be with us. And also I want to introduce Dr. Antonio Marcus Paes. I want to introduce him in Spanish because También tenemos aquí estudiantes, ¿no?, mexicanos y queremos que aprendan, además de inglés, el portugués. Entonces, Dr. Marcus Paes hizo su licenciatura en Ciencias Farmacéuticas en la Universidad Federal de Maranhão, en Brasil, una maestría en Biología Celular en la Universidad del Estado de Maringá y su PhD en Cardiología en el Instituto del Corazón de la Universidad de Sao Paulo, en Brasil. Dr. Paes es profesor asociado de Fisiología Humana en la Universidad Federal, también en Maringá, Brasil. Él es el investigador principal de Ciencias de la Salud en el programa de posgraduados y actualmente él tiene más de 40 artículos publicados en revistas de alto impacto en los últimos 10 años. Y, por supuesto, es el presidente del Brasil, DOHaD Brasil, y además es el presidente del Comité Ejecutivo para la organización del Congreso Mundial del DOHaD 2025. Thank you very much both for being with us. Thanks, and thanks to all the people that is connected right now. Thank you very much. Please, Elena, can you
share the
Yes.
slide, please?
Yes, of course.
It's for me a privilege to introduce Professor Lucilla Poston from King's College London in England.
Sorry, Elena, I have troubles with sharing the Fabíola, can you let me share?
What? The presentation?
Yes.
You don't need authorization for this. It's okay. Perfect. It's perfect.
Okay.
Okay. So Lucilla Poston, Professor Lucilla Poston. As Cristiane said, she is the president of the International Society for Developmental Origins of Health and Disease. She is the head of the Department of Women and Children's Health and of the School of Life Course of Sciences at King's College London. She holds the Tommy's Charity Chair of Maternal and Fetal Health. A graduate in physiology in the University College London with a PhD in the field of medicine. She directs a multidisciplinary team of scientists and health professionals, which she established in 1995. Her research team aims to improve the health of women and their children in the UK and globally by better understanding of the process which lead to complications in pregnancy, particularly premature birth, obesity, gestational diabetes, and preeclampsia. She has published over 300 peer-reviewed research articles. Professor Poston, a fellow at Endum of the Royal College of Obstetricians and Gynecologists, a National Institute of Health Researchers, Senior Investigator Emeritus, and Fellow of the Academy of Medical Science in the UK. She's a member of the UK Government's Scientist Advisor Committee of Nutrition. In 2017, Professor Poston was honored by Her Majesty the Queen for her service to women's health. So she is going to present today DOHaD research in the 2020s, approaches and challenges. Very welcome, Lucilla. I'm so happy you are with us.
Thank you so much, Elena, for that very kind introduction. And may I say, I don't normally wear hats with feathers in them every day. I'm very delighted to be able to be here to present to you. But just before I begin my lecture, I'd like to offer my congratulations to the team in Latin America for putting this meeting together and this course. It's an enormous amount of work. I know that because you've got an awful lot of lectures and it's over several weeks and you have to do all the assessments and everything else. It's really an incredible amount of work, and so well done, because you've got a lot of people to come and a lot of people will be much better educated and understanding of DOHaD and the life course at the end of this. And from my point of view as the president of the International Society, thank you very much. Because one of the things that we all aspire to do is to advocate the mantra of the society, which is to improve the health of mothers and their children over the life course. And that's what the whole of your course is about. So congratulations and thank you from us. Just for people listening to you, there's I think 80 people, just to give you one or two bits of information about the society. The International DOHaD Society has about 800 members across the world. We have now 10 individual regional societies, of which LA DOHaD is one. So that is how we made up. Mostly people in the DOHaD Society belong to a regional society as well. And the regional societies have their own programs such as this. And then the overarching society is the International Society, which I'm president of. LA DOHaD is one of, I think, the most active of our regional groups. So well done again for that, everybody, because it's a lot of work that you do all the time, apart from this course. And also, of course, they are organizing the next International Congress, which is in Buenos Aires next year. And so I hope that this course will really enthuse you in the field of DOHaD, and that you, if you're not a member already, will become a member of the society. And the way it works is that everybody becomes a member of the International Society, and you're a member of the Regional Society as well. So that's what everybody is a member of both. I mean, one Regional Society and the International Society. And that's the way we work. I'd just like to say also that I think DOHaD has never been more important. Never been more important. And I'm very pleased that most governments are now appreciating the quintessential importance of early life health. Starting in utero, in fact, starting preconception. Most people are now realizing that even beginning to think about health in pregnancy is too late for the next generation. We need to think about the developing gonads and the health of the embryo in early life. But the influence on later life health and disease is absolutely incontrovertible. And what is really important about it is also we're beginning to understand an awful lot more about the mechanisms, and that's what I'm going to be talking a bit about today, the methodologies, and that the world is now listening to us. But why is it important at the moment? Because we have a terrible pandemic of obesity across the world, which is undoubtedly having a major effect on the health of our offspring. Postnatally, overnutrition is a major problem in toddlers and school children. And on the other hand, we have terrible conflicts like Gaza and the Ukraine, where women's health is being ignored at the expense of everything else. So we have terrible conflicts. And then on top of that, we have climate change, which is changing. We've been doing a study in sub-Saharan Africa, and the drought in Kenya for the last 2 years, which now it's fortunately ended, has been terrible for mothers and their children. We've been doing a study over that period, trying to assess the influence of heat, the influence of drought, the influence of floods on pregnant women in disadvantaged societies. And that is going to get nothing but worse, unfortunately. So it's incredibly important that we as a community impress upon everybody that we need to look studiously at early life and the origins of disease later on, because it is quintessentially, as I say, quintessentially important for the health of the next generation. So this topic today is about advocacy, and I thought, well, what am I going to talk about advocacy? I have given talks about how we talk to government, and keep on, please, talking to everybody, policymakers. But as researchers, and which is what we know, I have a lot of postdocs and PhD students with us today, which is really good. As researchers, we need to advocate our subject. And what I'm going to be talking about is advocacy in terms of how we convince people about whether the methods that we're using are the right ones. Now, you might think that's obvious. We know that we should look at cohorts. We know we should look at animals. We know we should look at data. But it's not always easy to decide, if you're a PhD student and thinking about your future career, where you should go. How should we address DOHaD most effectively? And of course, we have to convince the funders who fund us to do our research that the methodology that we're adopting is the right methodology, because they won't give us the money otherwise. And we also need to understand the disadvantages and the advantages of the different techniques. Now, I am fortunate to have been around for a long time in DOHaD, because I've been coming towards the end of my career. But I have used animal models. I've used humans. I mean, I've utilized human populations to study the DOHaD hypothesis. I've done randomized controlled trials. I've done observational studies. And what I'd like to do today, having experienced all of those myself, is to give you a very high level, we don't have much time, but a very high level idea of what I think are the challenges and the approaches we should be choosing as we go through our careers in the DOHaD community. And you can argue with me afterwards. You can tell me what you think I'm saying is not right. But I think it's not always obvious when you're doing one particular area, such as you're focused on doing observational studies in cohorts and so on, about what are the advantages or disadvantages of the others. So that's what I'm going to do. I'm going to do a whistle-stop tour through the methodologies, and then I give some examples of them and some of the advantages and the disadvantages, and how you might think as you're taking your research in the DOHaD, early life nutrition or early life, whatever you might be doing, exposure to this and that. If you're a midwife, you know, what is the best way to deliver, about postnatal nutrition, about postnatal growth and all of that. I won't be able to go into much detail. So I'm going to share my screen now and go through my talk, and then we'll have some questions at the end. So I'll just do that first of all. And I'll share the screen, share that window. And can you see that? Yeah. Can you see that okay?
Yeah.
Yeah. Okay. So I've introduced what I'm going to talk about. How can we advocate to those people who matter? And that includes policymakers as well as funders, that what we're doing to investigate DOHaD and the early origins of health and disease is the right way to do it. So the approaches which I'd like to consider are, first of all, experimental animal models.
For me, it's not in the, sorry.
Okay, everybody, yeah. Human observational cohorts, randomized control trials, and then the disadvantages and advantages of using population level data, which you are extremely good at in South America. So I hope this will resonate with some of you who are working with the Brazilian cohorts or the Chilean cohorts and so on.
Lucilla, excuse me, Lucilla, I'm so sorry. Can you please just press the button at the bottom of the screen to share the not to share, but to put the presentation in full mode, in presentation mode.
Oh, is it not in presentation mode? Let me try again.
No,
I did press presentation mode, so I'm going to put it on there. I'm going to use slideshow. Is that right?
No, unfortunately not.
No, but that's possible to see. That's possible to see.
I'm sorry, but yeah, let me just have one more go then. It's actually working on my screen.
Yeah, I probably choose the full screen. Yeah.
Yeah, and then I'm using the full screen, and then it says use slideshow, and it's on slideshow for me, but not for you. No?
Okay, but it's for us. It's okay.
Yeah, I'm sorry.
We can see your slide with no problem.
I'm not
I
sure
guess
what we
you can go on. Thank you so much.
Sorry about that. When we did the test one, did it come up in the full screen?
Yes,
Earlier on?
yes,
Oh,
I think so.
Yes. But that's good to see. Don't worry. Don't worry.
I'm just going to try once more, and we'll see. So you can see my screen now, which has got, yeah?
We can share, Professor Lucilla, to share again and to choose the full screen in the options of the Google meeting. We can share. We can choose the.
Yeah.
Okay. Don't worry.
Is that good? Is that better?
One way, it's clicking on slideshow in the top. Slideshow.
Is that working?
Yeah, it's working, but it's not in the full screen.
No,
there's in the top animation slideshow. If you click in slideshow, we think we can change the.
I've done that. It's not working because it's.
Oh, sorry.
I've already pressed that. Yeah.
But don't worry, it's possible to see clearly.
That's a shame. I'm sorry about that. The typing is quite big. Okay, so shall we continue? Yeah.
Yes, please.
Okay.
Perfect.
So I'm going to talk about experimental animal models first of all. So let's think, you'll be more familiar with some of these than others. So So I've chosen a list of some which you will recognize. Now, for example, you're all very familiar with studies in rodents, probably, and maybe studies in sheep as well. But very simple animals, such as nematodes, fruit flies, can also be used in DOHaD research. One of the advantages is that they're very cheap. They're inexpensive. Chick embryos can be used, and I'll give you an example of that. And they've been used extensively in studies of development of different organs and so on. And we've understood enormously about the early development through studies of chick embryos, which is also relevant to DOHaD. And non-human primates, fewer of you will be using those. So we have quite a repertoire of potential animals to use, and each of them has their advantages and each of them has some drawbacks. So I'm just going to talk about nematodes for a moment, which might surprise you. But there have been over the years quite a few studies and impressive studies carried out in the nematode C. elegans. And this is an example from a recent study. Now, the good thing about this model, and I don't expect many of you have thought about using this model, is that it takes only 3 days to develop from an embryo to an adult. So this is much better than rabbits and rats and mice, which take a long time, and humans, of course, which take forever. But this model has shown very nicely, and there have been several papers on this, that early nutritional deprivation in the early developmental stages of this nematode is associated with abnormalities in adulthood, which of the gonads, which affect fertility. Now, I was looking at some reviews of the human studies and the other animal studies about early life nutrition and gonadal development and infertility, and there are lots. I mean, there is a substantive literature which suggests that early nutritional deprivation in mammals is associated with poor gonadal development, particularly ovarian development, and also that fertility may be affected. And it is also the case that testis development may be affected by early nutritional deprivation. So from that point of view, this model in this simple little nematode may be quite a good model of looking at gonadal development problems. So here is a normal control nematode, and when exposed to early life nutritional deprivation, it develops issues in the uterus and in the germ cells where tumors develop, and this is abnormal cell proliferation, which is what happens in ovarian tissue in mammals, and there is changes in the uterine cavity. So this paper is in the genetics journal, and rightly so, because this model is amenable to genetic modification through either knockout or knock-in, or RNA interference, RNAi, where you can make nonsense of a gene and it fails to the transcription is affected. And to cut a very long story short, and this is a very detailed paper, and I'd have a look at it if you have time. It shows that insulin and insulin-like growth factor was very important in this development of infertility and abnormalities in the gonads, and it was dependent on a reduction of Wnt signaling. Now, Wnt signaling we know about in cancer research, but it's also been implicated in abnormal proliferation of developing gonads, and has been implicated in quite a lot of DOHaD studies as a very important signaling pathway, which may be affected very adversely by early life adverse exposures. So that's just one example, but think about those simple models. Bacterial studies sometimes have helped with genetics, not so much. But, and as I said, the Drosophila, the fruit fly, has been involved in some DOHaD type studies.
Sorry, Lucilla, but I think the slides are not passing. Maybe I can share from here,
Yeah,
and you tell me when I can pass.
All right, then why don't we do that? So I'll take this off.
Yeah.
You've got my slides.
Don't worry.
Yeah, I'll stop presenting. You can.
Because it's very important.
That's why I sent them.
It's working now.
Okay, good. So if
Good.
you go down
Perfect.
to that slide,
Tell me.
chick embryos, next one. Yeah. Brilliant. Okay.
Perfect.
Not the one above, the one before that. Okay. So as I mentioned, chick embryos. So that is looking at just taking a hen's egg and exposing the hen's egg to different environmental insults. Has been used quite extensively, and one of the labs to have done this is Dino Giussani in Cambridge, and also he, as being a Bolivian citizen, has done quite a lot of work in the Andes, taking chick embryos in an egg to the top of a mountain or to sea level and looking at the effect, and it's about oxygen exposure and so on. And as I mentioned, it's the chick embryos. There are institutes which survive on studying the development of the hen in utero, as it were, in the egg, to understand the development of of an embryo, which is very, very much related to human development. So, very important model. So let's just have a look at one or two examples of chick embryo. Could I have the next one, please? So this is one of Dino Giussani's studies, in which they took eggs of a sea level mother. So a hen who was living at sea level up to altitude, that's on the left-hand side of the slide. And they showed that when those hen eggs were taken to altitude and the chick was born, as it were, came out of the shell at altitude, the chick was much smaller than it should have been. Conversely, if they took the egg of a high altitude hen down to sea level, quite the opposite happened. At high altitude, those eggs were producing slightly abnormally small chickens, but when that chicken was born at sea level, the chicken was bigger, and that is because the animal in utero in the egg is adapting to the high altitude, and it's probably through persistent oxygen tension and genetically modification over the centuries in terms of evolution. So that's a really nice study, and if you want to look, that's one of the early studies that Dino Giussani did, going up and down mountains with eggs, which I always used to tease him about. But can I have the next slide, please? And this is one of his most recent studies, only just published a few weeks ago. And this is showing very nicely that chronic fetal hypoxia in a hen's egg results in similar changes in the mechanisms which detect changes in oxygen, which include a system called the N-acetylcysteine system and the production of hydrogen sulfide. Now, that happens when an embryo is subjected to hypoxia. That system is activated and hydrogen sulfide is produced in the embryo. And it also happens that in this study, they looked at vessels from the umbilical vein and also from the placenta. And interestingly, the human placenta, interestingly, the same system was activated in those vessels as it was in the chicken's vessels. So this is telling us, if you're interested in the effects of hypoxia on embryonic development and the vasculature, which is what Dino Giussani looks at, he's shown long-term effects on the cardiovascular system of the offspring, that the hen egg is just as good as the human placenta for doing this study. And what he's saying is that this response, producing hydrogen sulfide, you may know, is a vasodilator, and N-acetylcysteine is an antioxidant. And what he's shown in this study is that, in the bottom half of that slide, is that N-acetylcysteine, NAC, if you put that into an egg, it stimulates the same pathway. So what he's thinking is that in utero, when hypoxia leads to fetal growth restriction in man or woman, and also in the chicken, that this is a protective mechanism, the activation of hydrogen sulfide, and it might be a treatment. So he's shown here that N-acetylcysteine improves vascular function, despite the fact that the animals are, or the egg is going to have a growth-restricted offspring. So it will be less growth-restricted than if it hadn't had this protective response. A very, very well done paper. It's incredibly detailed. But what it's also showing is, and you can look at the epigenetics of the DNA in both of these systems, and the same change in methylation status of the genes which produce N-acetylcysteine and H2S are affected both in the human placenta and in the embryo of the chicken. So should you be thinking about looking at hypoxia in people and fetal growth restriction, it's much cheaper to use hen's eggs than it is to use placental study. And actually, looking at human placental arteries and getting them fresh and placental arteries, as I used to do, and I have published many papers on that, is not easy. You have to be there at delivery, you have to pay somebody to collect the samples, you have to put them immediately in RNA later, you have to get them into ice. And if you don't do that, everything goes completely wrong and it doesn't work. So there's no point in doing it. Whereas it's much easier to control hypoxia in a hen's egg than it is in the human placenta. So that's just an example. You know, you might think, well, I'm going to have to go and collect loads of placenta because I'm interested in FGR. Actually, you might do just as well as taking a few eggs into the laboratory with you, and it will cost you a fraction of the price. So it's just, if you can think of alternatives, and Marcus was saying to me the other day, it's very expensive research, it's very difficult to get the money. So think of the cheaper options of using a good model. You have to justify your model. This paper will be a wonderful way of justifying your work in hen's eggs. So, and the funders would probably appreciate it, providing the evidence was there in the background. So let's leave chick embryos and think a little bit more about rodents. Next slide, please. So this is Elena Zambrano and I shared many intellectual discussions and discussions about papers and so on, and we've all been using rodents, particularly in relation to nutritional undernutrition or excess in the mother. And it has to be said that rodents offer huge advantages in many ways, because quite a lot of methodological tools are available in mice and rats that are not available in other animals. So, for example, you can get epigenetic arrays for mice now, whereas you can't do that for some of the other rodents. And of course, you can for humans as well. So that helps you match up. But of course, rats are born at a far more immature stage than that. They are altricial. We're altricial species, but we're less altricial than the rodents. Rodents have anything up to 12 pups, and we only have 1 or 2. And the placenta is, of course, very different. And anatomically, as I'm sure you know, the mammal variation in placental development and structure is remarkable. So there are big differences there. And if you're looking at placental function, it's not the best model. But whereas if you're looking at the effects of obesity in the mother, it's a good model. Because, I mean, and particularly for cardiovascular studies and metabolic studies, the basic metabolic pathways are not that different. It has to be said that in mice, lipid metabolism is quite a lot different, and in rodents from humans with very little LDL around. So if you're interested in oxidized LDL, it's not a good model to use. But I mean, these are the methods which we've used, just a few of them. Radiotelemetry, when you can, and Elena's used this too, when you put a probe into the upper femoral artery to measure blood pressure, to measure flow. The animal can be chronically instrumented and can run around in its cage and be unconscious, because, you know, putting an animal in, as we have on the right hand side, deliberately to create stress by putting it in an enclosed plastic tube, which is there on the right. That you don't induce stress in an animal which is chronically telemetered, and they run around quite happily. But if you want to look at the heart, you know, there are small animal methodologies for using ultrasound in mice and rodents now, which are fantastically good, fantastically expensive too. I think the price is not quite as much as it used to be. But we've been fortunate to be able to use cardiac ultrasound, and you can do all the things you can do in people. You can look at left ventricular function, you can look at ejection fraction, you can look at stroke volume, you can look at all of those parameters which you would normally measure in a person. So that's a huge advantage. And of course, and the thing that you can never do in humans, you can use a Langendorf heart preparation when you take the heart out of the animal and you put it in, you perfuse it, and you can look at responses to changes in ventricular pressure and so on, and ejection fraction. So we've done all of those, and a lot of other groups have as well. And there's an enormous amount of evidence now, and it varies between laboratories, but if the mother is obese, the offspring grow up with insulin resistance and themselves have increased appetite and predisposition to adiposity. Undernutrition, on the other hand, they also become overweight and have increased adiposity and insulin resistance as well. So the two models sort of converge in the offspring. But there's a massive literature on this now. And the reason that I started working in studies in obese pregnant women and their children was because I'd done this work. And because others had done this work, because when it goes across species, I'll show you a slide for work in sheep, you get very much the same pattern in the offspring and also in baboons and so on. So anything which goes across species gives you confidence. And that's another thing to do, is to look between species to see whether your model holds and is validated across the mammalian species range. And so it's good to do it in mice and rats, and perhaps in guinea pigs and sheep as well. Could I have the next slide, please? So this is what we found in the offspring of obese rodents, but so have lots of others. So this is these are reviews which are freely available, and basically there are some minor differences between different laboratories, but these systematic reviews essentially show that the offspring of obese rodents have poor cardiovascular function. So we've now done this in 3-year-old children of obese mothers, and we find very similar defects as assessed by cardiac ultrasound, and altered heart rate variability is something which is common between all these different models, and there's a sympathetic response which is activated and so on. So there are many similarities, and they're picked up very nicely by these reviews, which you might like to look at. But that gives you confidence in it being something which is real, as opposed to just being something you found in your lab. I mean, it's really good when other people reproduce what you've done, and that you reproduce what other people have done. So that's something else which is very important to put into grant applications, is validation of a model that you might be using. Could I have the next one, please? So if we are interested, like I have been, in developing interventions in pregnant women to improve or reduce obesity in children, which we've tried to do and failed in terms of obesity, what seems very interesting in this is that the interventions that we've tested in mice and in and in rats show that you can reverse the defects in the heart. And we've actually just published a paper this last year suggesting that although the children aren't much less heavy, their cardiac function is improved with an intervention. But that was a clinical trial with all the drawbacks of clinical trials I'm going to tell you about. I think it's right, but we have to validate it in a much bigger study. And this is the sort of thing, clinical trials, you might end up with very few children who you can study. And, you know, I'm quite proud of that work that we've done, but I wouldn't say categorically, hand on heart, as we say, that that is what happens in the population level, because the numbers were small and so on. So we have to be cautious about results of randomized controlled trials. So all of these studies here have shown that interventions in pregnancy can improve offspring cardiac structure and function. And Elena, who has done quite a lot of studies on exercise and so on, showing that metabolic function in the offspring and obesity can be reversed. So together, these interventions seem to work. So that gives you some faith across different laboratories, across different species, that interventions in pregnant women might work. But there are lots of reasons why they might not too, which doesn't anything to do with the methodology, which we'll come on to. Could I have the next slide, please? So what about sheep? So sheep obviously have a completely different placenta again, like rodents have a very different placenta from women. So they have this caruncular style placenta with lots of different caruncles around the main uterus. They're a good model in terms of gestation. They only have 1 or 2 offspring, often have twins, but they are a good model. They're incredibly expensive to house and to run. And the number of I mean, sheep physiology, fetal physiology that we understand now, I should say, fetal physiology, our current understanding is almost entirely due to studies of fetal physiology in sheep. And over the years, when people were beginning to understand the development of the fetal heart, the development of the fetal cardiovascular system, sympathetic nervous activity, skeletal development, and so on. There was a boom in studies of fetal physiology. The reason being purely that you can chronically instrument an embryonic sheep or lamb in utero, sew up the mother, and that lamb will survive. And then you can remotely record all of the variables in the fetal sheep. So our understanding of fetal physiology has been almost 100% initially, 100% derived from studies in fetal sheep. But having all of that been achieved, the studies in sheep now are dwindled across the world to almost very few units using them for DOHaD type studies. And the main issue about it is the cost. But some laboratories carry on. Dino Giussani in Cambridge has got a big unit where he's still looking at sheep, but it is incredibly expensive. But nonetheless, and of course they're long-term experiments, they have yielded enormous amount of information, which tends to concur with rodents. So what's the advantage of sheep over rodents? Not a lot in terms of understanding effects of nutrition. So I don't think they add an awful lot more, but they do give us greater insight into fetal physiology than is available in rodent models. So finally, non-human primates. Non-human, could I have the next slide, please? I, for one, oh, sorry, this is about sheep. That's just showing that the offspring of obese sheep, so they show the same sort of cardiac dysfunction as rodents, so why not use rodents rather than sheep? The next slide, please. So going on to non-human primates now. Next one, please. So I think if you spoke to anybody in the pharmaceutical industry, most people in the pharmaceutical industry, they will only be happy if you do studies in non-human primates, really, for new drugs, particularly in pregnancy. Not so much in drugs out of pregnancy, where you can transition from rodent studies to a first-in-man type study, certainly in relation to some of the immune function cancer studies and so on. But I've been discussing this quite a lot recently with a charity I'm involved with. It is difficult to progress to studies in pregnant women, and that very, very few new drugs get into pregnancy researchers' use. This is the major problem that we have, that very few companies or obstetricians will take the risk of a new drug in pregnancy because of the prior problems about diethylstilbestrol, about thalidomide and so on, and about the litigation costs, which are huge. So it could destroy a drug company producing a new drug for pregnancy, which may, for example, I mean, there's a discussion at the moment about semaglutide, and its use in pregnancy is currently contraindicated. But wouldn't it be good potentially for women to lose weight and not to have such large babies and to not get diabetes and so on? Nobody is going to take that step. It's also contraindicated in weaning as well, when it could be in postpartum. It could be obviously advantageous for postpartum weight retention loss. A lot of people are talking about this, and nobody is prepared to take the ethical challenge of using semaglutide in pregnancy. It's not saying that it wouldn't be safe, but we don't know, and this is the problem. So there is a need to do non-human primate studies if we're ever going to get drugs safely into pregnancy. Mostly drugs start being used in pregnancy when they've been used coincidentally, and people don't realize they're pregnant and they've had a pregnancy when they're still taking a drug. That is statins. There's a big series now on statins where pregnant women have taken statins without knowing they're pregnant, without any obvious disadvantage to the fetus. But we still don't allow the use of statins. They're still contraindicated in pregnancy, but there is an increasing body of evidence. And ultimately, they might be used because so many pregnant women have had normal outcomes when they've carried on taking statins. So to go back to this, obviously, there's a similar metabolism between humans and sheep, particularly in relation to lipid metabolism, for example. They have a discoid placenta. Placentation is very similar, and that is very important when you're talking about placental disease, fetal growth restriction, preeclampsia. They don't get preeclampsia apart from some indication in baboons that they might. So that is a dissimilarity. There is no good model of preeclampsia. be it in rodents or in non-human primates. Not a really good model. They have a long gestation. They have similar endocrinology of parturition, which is important. So, for example, they don't have progesterone withdrawal like sheep do and other animals. They don't have the corpus luteal production of progesterone that mice have, which continues pregnancy for a long time. So, for most of pregnancy. So they do seem to have similar endocrinology. And certainly in premature birth, it's considered that the only really good models of premature birth are in non-human primates, particularly the infection models and so on, but there still is not a perfect model. And the young are born in a more advanced state of development than the severe altricial rodent models, of course, so there are many similarities and so on. Again, hugely expensive, a huge problem about attitude towards the use of non-human primates in research, a lot of public opposition. And this is the discussion I've been having with the charity I'm working with. You know, we agree that in prematurity research, these models should be used, but to be able to get funders to agree to that is another matter. You have to have a very, very strong case. But they do have, and there have been some wonderful studies of non-human primates in the DOHaD field. Peter Nathaniel's in his group, particularly, and the group working on macaques in Portland, and so on, are doing very, very good studies, which to me, and the obesity studies in Portland, Oregon, have been very important. So they do give you ultimate confidence, not 100%, but pretty good confidence. So could I have the next slide, please? So I just want to say that there are some conditions where you can really choose your model, because the mechanisms are towards disease or whatever you're looking at are very similar. And I'd like to give you an example of this. So rodents, sheep, and non-human primates are all mammals. This next study, could I have the next slide, please, is a really important paper, which was produced by Horvath and his group and published in Nature Aging last year. And this is a remarkable study because they've looked at 59 different tissue types amongst 185 different mammalian species to look at what happens to your molecular mechanisms during aging. And what they have found in this study is that it's an extraordinary consistency in the aging clocks. You know about epigenetic clocks, which are indicators of biological aging as opposed to chronological aging. There's a big literature on this, and these are epigenetic marks which will give you a very good idea of the individual or the animal's actual biological age. So putting all these tissues together, and this diagram on the left shows it very nicely. On the x-axis, you've got the relative age. So they've basically averaged out the same degree of aging in all the animals. So between 0 and 1 for 0 for the youngest and 1 for the oldest. So they've basically adjusted for age. And then if you look at the DNA methylation for the relative age in all those animals, there's an absolute straight line. And what they've done is they've identified specific cytosines which are methylated, differentially methylated, and they're the same ones in all of these different animal species. They're exactly the same. And these are always in genes. It's the cytosine residues in genes implicated in development, which is not surprising, in cancer, obesity, and associated with longevity. So you could choose any tissue from any mammal, pretty much any mammal. I think there was one whale that was a bit out of kilter with this, the bow-headed whale, I think. And you could do very good studies on aging in any of those tissues from any of those animals. Well, that's an important discovery because it means that we can study aging in rodents, we can study it in marsupials, we can study it in any mammal, and you're likely to come up with the same sort of mechanism. Now, not everything is going to be the same, but in terms of, and these are related to mitochondrial function particularly, which declines with age. So it gives us a major insight into aging, but also the fact that we can safely probably use a lot of other mammals. So it is agnostic of the species, which is important. Okay, so now to move on. Next slide, please. I'd just like to say that the one, whatever animal you choose, and this is so important, we need to stick to and be adherent to the 3 R's principle of using as few animals as possible, to replace animals with other systems. So using simple animals like C. elegans is a very good example of replacement. I wouldn't say that organoids are really there yet with DOHaD research. Organoids, I have been involved with quite a lot of studies of organoids. They're very good. They're beginning to be very good for understanding placental function. But if we want to look longitudinally, as we do in DOHaD research, they're not really up to that at the moment. They will tell you about the cross-sectional. You could look, I suppose, at organoids from different stages in development, and particularly with guts and placenta, and they are good, but they're coming on. So, and some of the in silico type approaches are beginning to be involved. But I would have thought I would prefer replacement using a more simple animal, a cheaper animal, and one which is less controversial. So reduction in numbers, that can get to extremes when people don't have adequate power to do their studies. And I do worry about reduction in numbers a bit, because quite a lot of funding bodies are really insistent, particularly in non-human primate studies, of using the bare minimum. And that can be a real waste of money, because you can have, say, 10 animals in each group, which generally is not enough. But if you had 20, you might have the answer. So the power calculation for reduction has to be very, very carefully done, and you have to justify your numbers. But to go below what you think is sensible is also very dangerous, because it's a complete waste of funding. And I've seen that. I mean, that does happen. And people don't believe when you have an N of 3 that what you see is really true. But you've been made by the funders to use small numbers. So that's a warning about overdoing reduction and getting it right. Thank you. So for the next slide, I'd like to now talk about human observational cohorts in DOHaD, their use and their disadvantages, and how we should use them. So there have been an enormous number of observational cohorts. I've reviewed that subject area myself, and their promise of causality very seldom meets up to expectation, and which is worrying. So could I have the next slide, please? So I just put this together. So this is relationships between maternal nutritional status of one sort or another and childhood outcomes. So around the outer perimeter ring, we have the exposures which have been implicated in pregnancy with changes in neonatal and child health. So take an example with vitamin D. So there have been quite a lot of studies, observational studies, which have suggested that higher supplementation with just high vitamin D levels are associated with improved respiratory function in the offspring. There are a lot of studies, and there's some randomized controlled trials, but lots of observational studies, and suggesting that this, vitamin D in pregnancy might be a really good thing to take to improve childhood respiratory function. There are lots of studies of that. There are people who suggest that multiple micronutrients taken as a tablet, which obviously it's what's used mostly in developing countries now for micronutrient supplementation, is associated with improved childhood neurodevelopment. Definitely with improving birth weight, which it does a bit, but also further down the line in the DOHaD way, that associated with improved neurodevelopment. Similarly, with the obesity side of things and weight, a lot of suggestion that obesity in mothers is associated with obesity in children. And we've done studies, lots of people, the rat studies certainly would suggest that. And so on. Diabetes treatment can reduce adverse outcomes in children, particularly neurodevelopment and obesity. Those observational studies, and the other one that is really predominant is about omega-3 fatty acids at the bottom there, that supplementation or high levels of omega-3 fatty acids in mothers can improve neurodevelopment in children. So there are hundreds and hundreds of these studies. And to test this, I mean, one of the issues about these studies that you have to adjust for an awful lot of things. It's statistically, you have to adjust for the socioeconomic status of the woman. You have to adjust for smoking. You have to adjust for whatever you can adjust for, exposures, pollution, perhaps if you can. All these things which we know also affect these neonatal outcomes. That this modelling that people have done, and as over the years, as these cohorts have developed, the modelling has become much more refined. Longitudinal modelling methodologies and statistics are very important to understand and apply to these cohorts. When you can't assume causality, but you can get much closer to it by proper adjustment. But you can never really eliminate residual confounding. And I think this is one of the major problems of a lot of these studies. There is residual confounding. So the next slide shows what happens if you do randomized controlled trials to test, and the next one, please, to test these observational studies. And there are a lot of blank circles there. They're the ones that didn't work. So if you take multiple micronutrients, take maternal vitamin D, for example, the vitamin D intervention studies showed no improvement in respiratory function in the children, but some improvement in bone health, which was a secondary outcome. Maternal multiple micronutrients, there is no evidence from those intervention studies that you improve neurodevelopment, but there was a reduction in diarrhea in children, girls, I think, in one study. Omega-3 fatty acids, almost universally, there have been a lot of intervention studies, randomized controlled trials, showed no improvement in neurodevelopment in the offspring. Now this, you know, a lot of people, I mean, we all buy eggs with PUFAs in them and so on, very good for neurodevelopment, and the evidence from randomized trials does not match up to the observational studies. And it's worrying for people in DOHaD, like me, as president of the society, that this is happening, because does it mean that DOHaD's got it wrong? Does it mean that these associations are wrong, or is it something else? So we have to think very carefully about randomized controlled trials and what they might be telling us or not telling us. And this is again where I've had quite a lot of experience. So I'm going to have the next slide, please. Sorry, this is just an example of the association between maternal obesity and offspring obesity. Profound if you do observational studies. We've shown it, lots of other people have shown it, straight lines. If you do an intervention study in women to try and improve diet, exercise, and so on, this is a forest plot which shows that there is no improvement in obesity in the children. So what does that mean? That all the animal studies have told us it works, all of them. But if you do an intervention study in animals, it also reverses the defect. But if you do it in people, it doesn't. So that tells us something, and I have quite a lot of insight into what this might be. So so don't despair, DOHaDers, because I think this is because of clinical trial design and not because of, compliance and design are the two issues here. So if we could look at the next slide, please. So what is it about these intervention studies that means that on the whole, they don't work? Firstly, the participants who come into randomized controlled trials are not representative of the general population. So you can't always assume that's what's going to happen in the general population. They are self-chosen, motivated people on the whole, but then you might think they're the ones who would comply with the intervention, but that's not necessarily the case. More importantly, if you're doing a randomized controlled trial, you have to have a primary outcome. Now, funders don't want to wait until the children have got to the age of 5 as a primary outcome. Look at obesity at the age of 5. They would like to make the primary outcome an outcome at the end of pregnancy. So when I did the UPBEAT study, which is a study in obese pregnant women, I was basically made to have a primary outcome, which was gestational diabetes in the mother, because the funders wanted to know about that. But that means that your primary outcome, of which I would like to have been childhood obesity at the age of 3 or 5, becomes a secondary outcome. And secondary outcomes in clinical trialist definitions are not liked by publishers, and they're not appreciated by statisticians. And the reason for that is that there are a number of reasons. One is that the numbers change between the primary outcome and the secondary outcome. So if you look at the UPBEAT study, which we did, 1,500 obese pregnant women, when they were born, we looked at them although we had 1,500 births, of course, and we had very good neonatal data. When we looked at the children at 6 months, only 698 women would come back to the study. When we looked at them at 3 years, only 514, which was a third of the original population. We published all of this, and this is why I say when we look at the children's cardiovascular system at 3 years, we're not looking at the whole trial population, and that's bad news when it comes to validation and confidence in the results. If you look at any intervention study in pregnancy, the attrition rate is always very high. I mean, it's usually much higher than this. So that is a major problem about DOHaD research and using RCTs. The attrition rate is so high. So at 6 months, actually, we did show the children were a bit thinner. At 3 years, they weren't, but their heart rate was improved in the intervention arm, as I said, and cardiac function. But do you believe that? Well, if it's not representative of the initial population, first of all, you have to make sure you have enough in the randomized arm and the control arm of the study. But it is a major problem about these studies, a major problem. So DOHaD longitudinal studies are confounded by attrition, and also, and if you look at the adoption of the intervention by women in these studies, very seldom reported compliance. Compliance is pretty poor in some of the studies, so the women drop out. And when women drop out, you have to analyze the study by intention to treat, as we call it, and that leads to statistical issues. So the statistical issues are paramount. The attrition rate is huge, and I would say the biggest problem is probably compliance in the women. So although the women in our study did change their diet quite considerably, it just wasn't enough to change the outcomes. And we know that their gestational weight gain was marginally reduced. That's telling you they haven't really done very much. So this is the problem about designing clinical trials. You think that women will do what you ask them to do, and women decide otherwise very often. And that is always a big problem. So major issues about clinical trials in this field. And of course, it looks as if others will say, well, it's not working, DOHaD's not right. I don't believe it, because all of the animal studies show reversibility, all of the animal studies show mechanism. It is that I believe that the intervention is not the right intervention. We're not adequately changing behaviours in pregnant women. If you look at the literature, there is a huge literature on this now. So could I have the next slide? So I'm going to end up by doing what I'm doing, talking about what I'm doing now. And the reason what I'm doing now is because I've been through each of these different stages. And I am now, I would not abandon experimental animal models at all, but I've done that in obesity, and I want to move on to people. But there is an enormous amount still to be done with animal models. If we don't do discovery science in animal models, we are lost in this field. So we have to get mechanisms from animals or from in vitro studies. But please don't think that I'm saying that we shouldn't be doing animal models. I'm not. I've just personally been there, done that. I want to move on to human studies because I believe in these longitudinal effects of early life intervention. So what about population data? So it's increasingly used because the numbers are huge, and that's an enormous advantage. So could I have the next slide, please? So routine electronic health records are what people are using now across the world. They've become much more accessible. Certainly in the United Kingdom, we have the advantage we can link mothers to children because we have an identity number which links the two. They can be used now for intervention studies. So what we have real world trials, and also what we call time-interrupted series before and after studies. And some of those methods are increasingly used now. You can even do trials of drugs by matching patients within the population, and you have population representative participants. If you're doing long-term follow-up studies and you follow the mother to child, there'll be very little loss of to childhood follow-up, which is one of the major reasons I think this is why we should be using these studies for DOHaD. They don't give you nearly as much information about discovery and mechanisms. That's what the animal studies will tell you, and also smaller studies in human cohorts. I'm not saying don't do them. I'm just saying if we want to know what will happen at the population level, it is very helpful to use electronic health records and to intervene using the methods which are available. You can follow people up to adulthood, and it's hugely much cheaper than doing randomized controlled trials or observational studies where funders will have to pay you to do studies at the age of 3, studies at the age of 10, studies at the age of 15. Each point of follow-up costs a fortune because of all the staff. So obviously there are cost benefits. There's problems about missingness of data. There's problems about validity of data, all of that. There are issues about access and governance and ethics and all sorts of things. But despite all those drawbacks, I think this approach, not for everything, I think this approach is very attractive in certain, for answering certain questions. So next one, please. So I'm running a cohort at the moment, which is called Born in South London. And I'll just tell you a little bit about it to give you an example of what can be done. So could I have the next slide, please? So we have a core data set. This is called Elixir Born in South London. We have at the moment, this is compared my 1,555 women, we have 55,000 pregnancies in the cohort at the moment, which is a complete joy when you have been dealing with small numbers. We use what's called BadgerNet Maternity. That's our antenatal maternity record. We link that with our neonatal intensive care record. We link it with our mental health NHS data, and we link it with our primary care data. So that gives us an enormous amount of information about the mothers and the children as they're growing up. On the left-hand side, the linkages we add, we have what we call hospital episode statistics, which is a national statistical collection of when anybody goes into hospital. We get the record of that and why they go in. We've just acquired this last week health visitor data, when the people are looking at the children from 6 months to 2.5 years, so we're measuring the weight and the vaccination of the children. The pediatrics data we get from hospital records for the children, and we've also just getting access to national immunization data, which will tell us, and there's a real problem in pregnancy at the moment. I don't know whether you have that in South America, that women are not wanting to take vaccines, particularly after COVID. So we can look at accessibility. So this is very good for answering questions about management of pregnancy, which is what we're doing, what we call a learning health system, but also for looking at long-term consequences for the children. So when the children grow up in England, they are all tested at reception and at year 10 at the National Pupil Database. We also have a national pupil measurement program when their obesity is checked, and that's a really good program. HFEA is our infertility authority. We're getting pollution data, doorstep pollution data as we call it, and we're getting social care data from the courts. So the idea is to build up a holistic approach to maternity and to childhood health, and this is going to it does involve, and we're doing this now, enormous amount of statistical modeling, and we actually will be advancing to use AI in these studies because it's so complex. But it's very ambitious. Not many people have got such big linkages, but you guys in South America do have some extraordinarily good linkages that you can use the data for, and I've got an example or two of that. So just to give you an example of what we're looking at, in the next slide, a list of some of the, for example, we're looking at models of maternity care for women in areas of ethnic diversity and social disadvantage, and we'll be following up the children from those women. We're trying to improve maternal and child social inequalities by interventions with our primary care teams, and then following up the children. We're looking at the relationship between perinatal mental health and physical health. And we've got 27 different studies planned in this cohort at the moment. We just completed a study looking at the effect on maternal and neonatal outcome of having virtual consultations during pregnancy, which has suggested that the outcomes in pregnancy aren't as good as they had been previously. But that's the sort of thing that we can do, and it obviously has many applications in healthcare, but also for DOHaD. Could I have the next slide, please? I'm nearly finished. So I've known about this cohort for some time because I was involved in one of the funding bodies, which was funding one of the studies. But this is this, I don't know how you pronounce it, SINASC cohort in Brazil, which has amazing data. And what I was aware of was the studies which have been done looking at Zika virus in utero, and this is a sort of programming study, the effects on the children long term, which have been published. I think 20 papers or more have been published from this cohort looking at the effects of Zika. But most recently, I was looking at this study using the same cohort, linking to longitudinal postnatal growth data. And this is really a DOHaD study published in the American Journal of Clinical Nutrition by Rocha et al., looking at the growth trajectories of 336,000 vulnerable newborns. That's newborns either born prematurely or small for birth, and showing that their growth trajectories, particularly fetal growth restriction associated with stunting in later life, but divided into different subgroups. A beautiful paper published in a very good journal, and a real DOHaD study. So just to say, you have that, you know, in Latin America, you have that on your doorstep. I'm jealous. It would be lovely to have it. So what a resource, with every single birth in the country being well covered and doing that. So next slide, please. Just to give you an idea of what how you can use these health records for intervention evaluation. So, for example, smoking cessation would be one. I mean, that we didn't have these cohorts then to look at that in terms of fetal and childhood growth, but that would have been good to do. The sugar tax that you have in Mexico, we now have in England, and we are using, not us, but a group in Cambridge University have been looking at interrupted time series before and after intervention with the sugar tax in the population, at the population level. We in our group we believe it or not, haven't got folic acid supplementation in our flour yet, like you all have in South America. The British government is about to introduce that, and we'll be using our cohort and some other cohorts in the UK to look at evaluate that, not just for neural tube defects, but for fetal growth and other outcomes, neurodevelopment in children. So just to give you an example, the next slide shows that is the study looking at primary school children after what we call the soft drinks industry levy, that's our sugar tax, an interrupted time series analysis done in the University of Cambridge, showing a trend towards increasing obesity. This is important to look at trends in school children, and that after the sugar tax was introduced, that trend went right down, only in girls and not boys, which was interesting. It's probably something to do with the level of puberty and being reached, we don't know. But they've also published a really nice paper showing that dental caries, extractions for dental caries have gone right down in these children since the introduction of this sugar tax. And they've also shown that asthma has gone down in relation to the reduction in obesity, because obesity is linked to asthma. So this is the sort of thing we can do before and after, but as I said, we can also do real world trials, of which I have no experience yet, but would be very interested to be able to try and do that. But any intervention, population health intervention, these health records in terms of DOHaD offer enormous potential. So I would really recommend, if you're interested in data science, to start thinking about this approach and convince your funders it's a good way forward. So next one, please. So this is my last slide. So researchers in DOHaD need to be advocates for the method they want to use in DOHaD research. So in animals, you should choose an animal model which really faithfully mimics the human condition, and which you can justify with an evidence base from papers which have been published previously. And what a condition you're looking at could be completely different for a different animal model. So you've got to choose your animal model carefully. Look at the mechanistic tools which are available for the species, because you can't do really high-powered transcriptomics or in epigenetics in some species where you can in others. And these days, that's very important. It's also expensive. So to look at the cost, it's important to choose the least expensive appropriate models. I'm suggesting if you're looking at fetal growth restriction, you could use hen's eggs in relation to vascular function, but maybe not in relation to lung size or brain development. You know, so you could only use that in relation to vascular function, but it looks like a good model. So look at the literature, really understand the literature, But above all, remember the 3 R's. But as I said, don't underestimate the sample size, because you can end up in a real problem. So I hope that's made you think a little bit about the approaches that you can take in DOHaD research. All of them have their benefits, all of them. Some of them have disadvantages. But if you're thinking of, could you just get the next one? Sorry, the next slide is about human studies. One of the most important things about human studies is, you've been working in animals, don't think that you can do studies in people, because it's a whole new world. I was very fortunate to be working in an obstetric department with a lot of people who could help me work with people, and I had a baptism of fire, and it was difficult, but I had a lot of people to help me. You can't suddenly start doing it if you don't have the skill base, and if you want to change over, get some training. I have to do clinical trials training routinely, and I have to understand data, human data. So I have to take courses to do what I do, and you have to retrain a bit. If you're doing observational studies, think terribly carefully about confounders, and when you're designing those studies, make sure you collect the information about all the potential confounders, which of course makes the study more expensive, because you need to have bigger questionnaires. Use really good statistical modeling, always have a good statistician. RCTs, I've gone right off them in terms of DOHaD, as you can tell, because I think they're flawed in many ways, but the intervention design is just equally important. So if you are doing an RCT, make sure you have an evidence-based intervention which is likely to work. And we have shown that behavioral studies don't work very well. Don't do them. Think of something else. If it's semaglutide, then you've got to persuade somebody to do that, but preconceptional studies is what I would recommend, and that's where there's a lot of emphasis at the moment. The more difficult to do. So consider using population data, whether it be observational or interventional. The opportunities are there. The disadvantages are the quality of the data very often, and if you've got really a lot of missing data, then you're not going to be able to do it. But you've got to look very carefully. And there again, I've had to join a bunch of data scientists. I had no idea about data science. I'm still being taught by them. I can tell them about the academic side, but I rely on the data scientists to do the work. And it's a different world completely. It's a very interesting world, but it's a different world. But don't be frightened about it. It's a lot of jargon in it. Get into it. Get a course, go and do a master's course, whatever is required. You can do it. It's not that difficult, but it's just a different language, and it is off-putting. Okay, so that is my last slide. And the next one is just to thank some people who contributed to my work, particularly our funders. That's Medical Research Council of Great Britain, the British Heart Foundation, and the European Union, when we were in the European Union, but we aren't anymore, and our NHS research funders, the National Institute for Health Research. So thank you. I've gone on for quite a long time. I hope this is over an hour, I think. So I hope we've got some questions that I'm very happy to answer.
Thank you, Lucilla. Great lecture. As you can see in the screen.
Thank you, everybody. Thank you.
So this is a moment of questions. People, feel free, please, to ask in Spanish, in Portuguese, in English. It doesn't matter. If it's in Portuguese, Fabíola or Cristiane can translate it. If it's in Spanish, Erika is in charge to translate it. But we have a great woman in science, that is Lucilla. Let's take the opportunity to ask. So in the meantime, until we receive the first question, I will start with the first question. Lucilla, very interesting data. And I know that in the animals models, there are many experiments between the difference between male and female offspring, the psychomorphisms, but I'm not very clear about the epidemiological and clinical studies.
No, there are a few, and we have shown some in our small studies of people from randomized trials. We have shown some male-female differences. Particularly, one example is heart rate variability is different. The heart rate is different. between offspring of obese female offspring of obese mothers and male offspring. So there are differences. You're absolutely right. It's hugely under-investigated in the animal models. And if anybody's using animal models, please look at the mechanisms of the difference, because they do exist in people. But of course, with the big data studies, we will always divide the children into males and females, and we will have thousands of them. So hopefully we will get some better indication in relation to maternal exposures. That's going to start off being observational. But we will have very large numbers. But the important thing with human studies is having the confounding data. You know, if the mother had preeclampsia, if the mother had diabetes, you have to adjust for all of those. The deprivation, indices of deprivation, BMI of the mother, if you're looking at something else. You do have the advantage to having a lot of data, but you don't have it all. So you'll still have a problem about residual confounding in the human studies. But yes, everybody should look at males and females. Very important. I don't know what it's about. I don't know what I really have no idea why.
Well, even in animal studies, there used to be more studies about males offspring, and females, we are very different.
Nearly all originally the human studies looking at drugs were all based on male subjects. Mm-hmm. And now there's a literature showing that males and females react differently to drugs like antihypertensives. But all of the original studies were done in men.
Mm-hmm. So this is not only women in science is important, it's also the kind of science that
gender medicine is called, very important.
Yep. Okay, before I ask the second question, how can I check who is asking questions? I don't see in the chat.
I'm following Professor Elena.
Okay, so Fabíola, are you going to?
Don't worry. I am fine. I'm reading the tests. We don't have questions in the chat, but if somebody would like to do some questions, se alguém quiser fazer uma pergunta, por favor, levante a mão, faça a pergunta, abra o microfone. I am available. Professor Dan Saladino stay with us now too, and vamos aproveitar aí. Thank you so much, Professor Saladino.
Please don't hold back. Anything, I can answer anything. Paulo, hello, Paola.
Casanello, Paola.
Hi, Lucilla. I was supposed to be there at the beginning, and I was in a meeting, so I'm just going out of the meeting, traveling back home. I'm very.
Good to see you.
Can you hear me?
We spoke to spoke to each other last night.
Yes, we've seen each other very frequently lately. I had a comment regarding. Do you have any knowledge of anybody looking at pregnant women regarding if they're pregnant of a female or male fetus? Not only the baby itself, but what the fetus, the sex, the biological sex of the fetus is making in the mother. Because
that's one thing to adjust for is for anything in maternal in pregnancy is the sex of the fetus, and there is, I mean, boys tend to be smaller than girls anyhow, so there is that known, but people are increasingly doing it, but there's not much work on it. But I was talking to somebody the other day who said, you know, what is the sex of the fetus? Are you adjusting for it? And I think that's something you should adjust for. We don't know why, but there are differences. Not many outcomes I'm aware of, but I think it's a good policy to adjust for the sex of the fetus. But yes, it's a good question, Paola, because I'm not really aware of much work on it, although people are aware that boys tend to be smaller than girls, and therefore mechanisms might be different for fetal growth restriction, because boys tend to be less resilient and smaller, so more likely to die if they're fetal growth restricted. It's a good question.
Any other questions?
We have one question here in the chat. It's from Renata, from Sarah, in fact, the first one. We have two. Sarah asks about an article that you show that correlates maternal obesity and cavities in the babies, in the child.
So, but obesity and
cavities in the
dental cavities, dental caries, yeah. So the in the United Kingdom, we've introduced what we call a sugar tax for sweetened drinks only. This is like you've done in Mexico. And it has reduced the sugar content of those sweetened beverages by 34%. I should have explained this more. So it's a big effect. And so this group in Cambridge looked at the number of operations for removing childhood teeth because of dental cavities. But that is the commonest operation in pediatrics in the United Kingdom, is removing children's teeth with general anaesthetic under the age of 5. It's the commonest operation. It's terrible because they live on sugar. So this is terrible. This sugar tax has reduced substantially the number of operations for dental caries. I mean, again, there are people saying, well, you haven't adjusted for this and that, and it was going down anyhow. But there's a dramatic effect after the introduction of the tax. And it's not surprising if the government has worked out how much less sugar in grams the children are eating every week, and it's really quite substantial. So it's but the issue about this is that children don't get most of their sugar these days from sugar sweetened beverages. They get it from top of the list is yogurts, confectionery, cakes. So sugar sweetened beverages actually is quite low in the list as their source of sugar. So what we are hoping is, I work on the Government Committee for Nutrition, who's really supporting this, is, well, I'm supporting it, but I work on the committee that's produced the evidence, is that we should extend this tax to these other sugar containing products which are higher than the list, particularly yogurts. It's extraordinary. But so they've created a list in this report that we did of feeding children from the age of 1 to 5. It's worth reading. It's this SACN, the Scientific Advisory Committee on Nutrition. There is a list of what children get their sugar from, about 40 different products. So sugar sweetened beverages are not at the top. So we need to, with our new government, we have a new government in England, thank goodness. We are hoping to persuade them to to expand the sugar tax. Personally, I think this is the only way we're going to combat obesity. Because
I believe you.
Yeah, it has to be top down. If you look at, I mean, what's happened with smoking in the UK, it was the government that banned smoking, and then basically banned smoking in public places, in restaurants, and that worked overnight. And this people do not adhere to behavioural change. It's largely, in my mind, because the food industry has got such good marketing for unhealthy food, and unless we tax the food industry, which is essentially what we're doing, we will not reduce obesity across the world. I'm very much sold up to that. So my personal opinion, it's not opinion of the committee I'm on, but it's my personal opinion. But having seen the evidence over the years, and having done an intervention study which didn't reduce obesity in pregnant women, despite an extraordinarily well conducted intervention with health trainers and people who knew what they were doing, and all sorts of evidence-based ways of behavioural change we chose, it didn't work, and it doesn't work, and it doesn't work across the world. Every single study has shown really, I remember systematic reviews suggest there may be some reduction in GDM, but actually, if you look at the individual reviews, there's no intention to treat follow-up. The statistics are a lot flawed. The big studies, which were well done, show nothing. So it's not going to work, and it doesn't work for the general population. That's not just in pregnancy. Children may lose some weight from intervention studies temporarily, and then they go back to their original weight. So that is, semaglutide is not the answer to obesity. It is a horrible way to treat obesity, but at least it does work. But we need to reduce the sugar and fat consumption in our food.
It's perfect. I think, Professor Elena, Professor Lucilla, we have just one more question in the chat, and after that we will finish. Yeah. The question is Renata Fabiani. Hello. In addition to the issue of RCTs, we also have the issue of the legislation in each country regarding studies on human animals, correct? which can be the cause of data confusion?
Which what? Sorry, last bit. Which?
I'm sorry. Which can be cause of data confusion about
Yeah,
this
I see what you
test?
mean. Yeah. Well, it's a nightmare. The legislation in human studies and the governance and the requirements for using data are so complex. It's a great shame because it does hold up using. I mean, it took me 3 years to get the permissions to use routine electronic health record data, which is ridiculous. If you speak with women, which we do, they say, "Why on earth are you not using it already?" You know, I have no objection, but we're using access to data, which is really opt-out consent, as we call it. So we have to go through a whole bunch of legislation to do that. Clinical trials, it's becoming utterly almost impossible in the United Kingdom to get clinical trials going because of legislation. So I understand it, and you have to close all the loopholes which there may be, but unfortunately, if you want to do clinical studies now, there is a massive amount of legislation you have to go through, and it's destroying people's confidence in being able to do them in the United Kingdom, which is a great pity. So yes, I agree, it's a real problem. The legislation certainly prevents you from being attacked legally, which is very important because there are increasing laws about data protection and so on. So we have to obviously adhere to all of that, but it makes life very slow in getting things done. So I agree, it's a real problem, but every country is different. We do adhere to European legislation, even though we're no longer in the European Union. So it is pretty much the same as across the whole of Europe, but it's still very difficult. Thank you for that question. It's very important.
Thank you. Lucilla, we have several questions, but we are short in time at this moment.
Yes.
I think that Gabriela, that will be the last question, and the other that we have in the chat, we will send it to you by email.
Please do.
Because I don't know, there are many. I'm so sorry. Your talk was very good. So, Gabriele, what do you want to ask?
Boa tarde, eu gostaria de pedir a ajuda dos professores com a tradução. Eu gostaria de saber quais que são, assim, se existem, né, desafios, assim, dificuldades na implementação das intervenções baseadas no DOHaD na prática clínica.
Professor Lucilla, the question is about the interventions. What are the challenges to interventions in clinical practice when you are thinking about the DOHaD concepts? I think it's something like your study, the upbeat, that you have a complex intervention.
Yes, a complex intervention.
Yes, exactly.
So the challenge is to get a really good intervention. I mean, when we did that, it was 10 years ago, so things have changed. We understand more about the exposures. So what we did a complex intervention, which was behavior, diet, exercise. We used changing behavior structures and from psychology, and we delivered the intervention in a way which was evidence-based. The intervention is we have to understand that, for example, obesity is associated with socioeconomic deprivation and poverty. And that is something which, if you change, would probably reduce obesity very effectively, but it's extremely difficult to change deprivation in an intervention, but it's a critical determinant. So the intervention should include some attempt to reduce inequalities, which is really hard. It should include some better appreciation of nutritional value, more education, much more education. But that is also difficult to impart over 9 months of pregnancy. So that is where preconception interventions may have better effect. You have a longer time potentially. I mean, in anybody of reproductive age. So what you're talking about is education in schools, to my mind, which has to come along with the top-down tax to the commercial sector. But a combination of the two, reducing accessibility to food. We have a lot of fast food outlets all around our schools, deliberately around our schools. You know, McDonald's and all of the Kentucky Fried Chicken, they're all around our schools. There's no legislation against that. So that's part of an intervention. If you're doing a population-based intervention for children and their parents, you need to get at that. So it has to be what we call a systemic or holistic approach to the problem, which appreciates all of the social determinants as well as the biological and nutritional determinants of obesity in that case. And the same applies to any other behavioral intervention. If it's mental health, for example, your mental health intervention should encompass all of those different aspects, social, biological exposures. And it's extremely difficult to do. And it's very expensive, but people are increasingly making an intervention more and more complex. But it's better to do it at the population level when you improve the environment, you reduce deprivation. That's the way to do it. It's the government that has to do it. It's very difficult for academics to do all of that. So you have to work with government, you have to work with policymakers to design community-based interventions, which may include antenatal care as well. Difficult.
Wow. Thank you very much, Lucilla. Great lecture. And one important message that I got from this lecture is that Latin America is a good land to make studies, epidemiological studies, and we as a researcher, we can try to find ways not to do really crazy expensive experiments.
Yes, exactly. And I think you have a wonderful opportunity with all of your birth cohorts and registers and so on. And if I were you, I was a PhD student, I'd go off and get trained in data science and maths.
Okay,
as well as doing all the biology. So you have to understand both of them, but it's a good thing to do.
Yeah. Well, thank you very much. And well, we will have the break for this course. It's 10 minutes. 10 minutes from now. I cannot say the time because it's different in every country. But see you in 10 minutes. And thank you.
Thank you very much, everybody. I'll say goodbye now then. Thank you very much. Very nice to see you all again.
Thank
Bye-bye.
you. Thank you, Professor.
Good luck with the course. Bye-bye.
Thank you
Bye.
for a lovely talk.
Bye-bye.
Many thanks, Lucilla. Bye-bye.
Bye-bye.
That's perfect. We can return in some minutes. Professor Dan is still there. Hello, Professor. Oh, how are you? Thank you so much to stay with us, Professor Saladino.
That's okay. Can you hear me okay?
Yeah, perfect. We can try to share your presentation too. Yeah, I think.
Do I click the present now button? Is that how I do it?
The button between the hand and the smile face.
Oh, okay.
The better option has full screen.
Okay. Can you see anything now?
Just a minute. Share did it appear a button, blue button, share something like that? Yeah, there are 2 steps.
Okay.
Oh, perfect. Perfect. Now we need you to see, I see your computer, but I don't see your presentation. I don't know if you share one page or full screen. Don't worry.
Okay, let me
we are seeing video calls and meetings for everyone. Yes, okay. It's this page. Maybe it's the other one.
Okay, let me
don't worry.
So if I have the PowerPoint.
Yeah.
And then I have this present now button between the smiley face and the hand. So if I click and then, okay. Right. Let me see.
Don't worry. The secret is share full screen.
Entire
not
screen.
One page. Yeah.
Okay. And
it's a small secret.
if you're using two screens, sometimes you need to push your presentation from one screen to the other.
Right.
Yep.
I'm just using. I'm using one screen, but I'm using two tabs.
Yeah.
Let me try again. Sorry about this.
Don't worry, don't worry. You have 5 minutes.
When it says choose what to share with Google Meet, I click on the entire screen.
Yeah,
Screen.
this is an option. Then we see your screen. Or I think it's the best option. If you have only one screen, I think it's the best option.
Yeah.
Yeah, exactly. It's the best option because exactly.
So if I
Some choice.
I think there might be screen preferences. Sorry.
Some choice. We have
select a tab to share. So I'm just wondering why it's not. So entire screen. But why is it I can't see my PowerPoint? That's the problem.
If you want, you can send the presentation to us and we can open from here. No, it's not a problem. We can open. Fabíola, do you have the email from Fabíola?
Yeah, I have received the presentation.
Let me just try one. Let me just try and change this setting.
Fabíola, do you want that I helped you? You don't need help?
Me? I'm sorry.
Yes?
No, I think everybody is inside now.
Okay.
And Saladino left the room. He's going to try to come back.
Yeah. We have 5 minutes. Is a break to go to the bathroom.
That's why I was asking you. You don't need something?
We start. There is a small secret here because we have 3 options. Let me see. One guide, the window of full screen. The best is full screen.
It's the easiest, I think, yeah.
Yeah.
But then you have to have the PowerPoint open and in full screen to show it. Dr. Dan Saladino, it's your microphone is closed now.
Sorry.
Yeah, now it's up. It's working.
Yeah. Okay. Screen.
Yeah.
Yeah, nice. Perfect.
Okay. So
yeah.
Then
it's not in full screen now. You can click on the button.
How is that looking now? Does that work or no?
Yeah, it's working, but it's not in full screen yet.
Oh, okay. Let me go back to the.
Don't worry.
No.
I think you can go back to PowerPoint. In your computer, it's showing full screen now, the PowerPoint.
Yeah.
Okay,
well,
let's change. Sorry. We can change the slide presentation. There's 3 options there, but we are not seeing your screen now. If you can share with us again. Then enter in PowerPoint. Yeah, there's a slideshow. Let me see. It's maybe presenter view, I think.
If I try presenter view.
Okay. So.
What can you see?
We are seeing your presentation, but not in full screen, but we are seeing.
Did you try play from start?
Yeah, I've tried play from start, and this is now in
so custom
presenter
show.
screen.
Custom show. Let's try there. Custom show, probably there are options.
Okay. Custom show?
Custom show, yeah. Let's see.
What happens when I click custom show?
For us, nothing. For us, nothing.
No. Maybe if you play from your end.
Yeah.
Sorry about that. I'm sure.
No problem.
I just need to practice. Yeah. There's a setting that I haven't quite changed. I think it's when I.
Sorry. Are you using two screens? So you could make the one you want to present screen number.
I'm using one screen.
Oh, that might be the issue.
Right.
Thanks, Aro.
Yeah.
Thank you.
I
can share
when it
for
says
you.
choose what to share with meetgoogle.com, it says Chrome tab, window, or entire screen, or it says select a tab to share.
I understand. Since it's one screen, so it's going to be sharing that screen unless you want to make it such that you are presenting but you don't see us. So that might be like you share the entire screen, we see, but you can't see us.
Are you okay to share at your end?
Yeah.
Yeah, sorry
for sure.
that. Just to make sure that it works. Otherwise, we could be here for. So you can see me, and then I can get the same version of the PowerPoint up, so I know what's coming. And then if I just say change slide, is that okay?
Okay.
Yeah.
Perfect.
Yeah.
I can share for you.
Okay. Thank you.
I can share for you, and I just do your presentation, and after that I closed my microphone and share your presentation, and you talk me next.
I'll just say next.
Together.
Yeah.
We are together. Thank you so much, Professor Dan.
That's okay. Thank you.
Let's go. We can start. Yes. Very nice. Very nice. Hello, everyone. Thank you so much to come back with us in this very important day. Today, I have a great pleasure to introduce you, Professor. I don't know if you are a professor, but for us, you are a professor because you're teaching people. You teach people, you are a professor. Professor Dan Saladino. Dan Saladino is a food journalist, broadcaster, and author of Eating to Extinction, the World Hardest Foods and Why We Need to Save Them. It is a very nice pleasure because I sent a mail for Professor Saladino 2 or 3 months ago to introduce our discipline, our course, and he replied me very fast and say thank you and say yes, I will be with you to talk in your not in mine but in our discipline. Here I have just to share with my audience one test in New York Times about the book of Professor Dan Saladino reading for Molly Young in January 25, 2022. And there is a small phrase, Professor Saladino, in this test that it was very important for me to start to follow on the internet, Instagram. I will do this. I will talk about that, and after that I will translate. What Saladino finds in his adventures are people with so deep relationship to their food. This is not the decadence or the preciousness we might associate with a word like foodie, but a form of reverence. Em português, o que Saladino encontra em suas aventuras são pessoas com relações profundas com a comida. Não se trata da decadência ou do preciosismo que poderíamos associar a uma palavra como food, como comida, mas sim uma forma de reverência. So, thank you so much, Professor Saladino, to stay with us. I will share your presentation, and a great pleasure to stay with you in our meeting now.
Thank you very much, and thank you for inviting me to join this conversation. I am a food journalist. I make primarily radio programs for BBC Radio. And for the last 15 years, I've specialized in telling stories about food and farming from around the world. But also, in addition to my BBC work, in 2021, I had my first book published, which is called Eating to Extinction: The World's Rarest Foods and Why We Need to Save Them. And the subject of the book and the theme of my talk is about agrobiodiversity. So it's about agricultural, biological diversity. So what I became really interested in is the diversity of different plants and animals that humans have interacted with for thousands of years in different parts of the world, from different ecosystems, and how that diversity came into existence, why it's been so important to our evolution as a species and our interaction with the planet, and also why in more recent times that diversity is disappearing and why we should care. So if you move on to the next slide. So in this talk, I'm going to be talking about food diversity, just explaining what that means to me, and also some of the stories that I've found as I've traveled around the world to understand food diversity. And also, I think the importance of storytelling. So I'm a journalist and a broadcaster, making radio programs and writing books. And I spend a lot of time working with academics in different parts of the world who are doing extremely important work about biodiversity, about food systems, about nutrition. Much of this is not finding its way into the mainstream. And so my responsibility, or my role as a journalist, is to be a storyteller, a communicator of that research and that work, which is why I want to make radio programs about food politics, food science, and also about food history, anthropology. And I also want to try and inspire people to understand why it's important that we all know where our food comes from, not only in a sense of geography, but also in terms of the thousands of years of humans farming, and before that, our time as hunter-gatherers as well. So if you move on to the next slide, I will explain the origins of how I became so interested in food diversity. So I, very early on in my career as a food journalist for the BBC, was lucky enough to travel to Italy to attend some events that had been organized by the slow food movement, which had been set up in Italy in the 1980s. And an important part of the rise of slow food and its global projects as an international movement was to look at traditional foods and foods that reflected a place, a region, a community. And part of what slow food do in Italy is to bring people together, food producers, farmers, from different parts of the world, to talk about the food traditions of that place. But so concerned was slow food in Italy of the disappearance of flavors of vegetable, fruit varieties, animal breeds, even recipes, that it created a project called the Ark of Taste. This is like the Noah's Ark of Taste. So a way in which they wanted to raise the profile of some of the things that were, or the foods that were disappearing from Italy's regions, with fast food arriving into Italy and replacing a lot of the traditional foods. So the idea was that if you can tell the story, if you can create a way in which people have a platform from wherever they are in the world to tell the story of the food of their place, at least we know that food exists. And if we know that food exists, we can understand its story, and perhaps we can all do something to try and save that food from extinction. And the project started in Italy, but then became a truly global project, covering 150 different countries, with more than 6,000 endangered foods. And if you go onto the Ark of Taste website, the Slow Food Ark of Taste website, you will also be able to see foods from Brazil that have been added to the Ark of Taste, and it will be broken down into different regions, which is obviously in certainly in the case of Brazil very important because of the size of the country. But it's fascinating to see the amount of diversity that has existed around the world, including Brazil, and each one of the foods has been included in the Ark of Taste because somebody around the world wants to save that food and is concerned. And it could be a food producer, it could be a farmer, it could also be somebody who is a member of a community and wants to save their food history and their tradition. If you move on to the next slide, please. So I was completely inspired by this catalogue of thousands of endangered foods. Each one of these ingredients captured a story of the development, the evolution of that particular food, whether it was a type of wheat or a type of rice, a breed of pig, for example, a type of nut, which could have been used or is still being used by indigenous people, or maybe has had a more recent development and has become important to that part of the world. But the key criteria for the Ark of Taste is that it is specific to a part of the world, that there is a history that can be told, whether oral or written, and also that it has a place within the culture and the community. So, for example, in the radio programs I made telling the stories of the Ark of Taste, which you can still find online through the BBC, there are types of peas and beans that you find in the west coast, on the west coast of Africa, that might be a story told by someone in the southern US, or in fact in Brazil, because of the movement of people over centuries, and particularly in terms of enslaved people who traveled with some of the seeds as well, or from Africa, arriving in the Americas, and it becomes a really important food with an important human story, a part of global history as well. So as a food journalist, the Ark of Taste became a treasure trove, because even a tiny pea could capture hundreds of years of history of how foods move around the world. If you move to the next slide, please. So I spent time traveling around the world, making radio programs about these endangered foods, becoming obsessed in a way with these stories. And it led to three main questions for me. So how did so much diversity exist around the world? So why is it that we end up with such different foods in different parts of the world? Secondly, if these foods are endangered, why? And the most important question for me to communicate to a mainstream audience, whether it's through a BBC radio program or my book, is why should we care? Why should we care about this diversity that has been so rich in our history as species, as Homo sapiens. Why is it disappearing? And that important question, why should we care? So next slide, please. And I became very interested in the origins of where did our food come from. And as you can see here, this map, the so-called Vavilov centers of plant diversity and the areas of origin for agriculture. So in the 18th and 19th century, there was a lot of interest in starting to try and explain what were the origins of some of the most important foods that were feeding huge populations around the world. So where did wheat come from? What were the origins of rice and maize, for example? Which is why we end up with this map, which certainly started to really take shape because of the science that was available of being able to look at the amount of biodiversity of different plants around the world, so we could start to map where did we find the richest diversity of a particular plant, and where the richest diversity could be found, it was usually where that plant had originated and had time to adapt, to mutate, be selected by humans to create huge amounts of diversity. So this map illustrates the centers of origin. And let me go into a bit more detail in the next map. So, for example, this is the Fertile Crescent. And this shows what we often describe as the Middle East. So the Fertile Crescent is you can see this arc, this crescent shape that covers Syria, Iran, Iraq, Israel, and also through, as you say, they're heading right into Jordan, and so that part of the world is where the so-called Neolithic package originated, and the Neolithic package consists of wheat, barley, chickpeas, lentils, and a type of leguminous plant, vetches. And these originate in different parts of the Fertile Crescent and spread out around, starting around 9,000 years ago. So what we have here is an example of humans interacting with wild grasses or members of the grass family, mostly, particularly with wheat and barley, and selecting, so-called process of domestication. And we see the human selection starting to change the wild genetics into what then becomes an agricultural crop. And as we now know, particularly with wheat and barley, they spread around the world, and from the so-called Old World to the New World in the Americas. And if you move on to the next slide, it wasn't just exclusive to the Fertile Crescent. In China, in river valleys, we also see humans interacting with another type of wild grass, which becomes rice. Again, we're talking about 9,000 years ago, humans selecting for the plants which give the biggest or the largest amount of grain. They're the ones that are the easiest to harvest. So humans select and they select and they select until they end up with something that is a cultivated, domesticated plant that the humans become dependent on, and the plant becomes dependent on the humans. So we see this interaction between humans and plants. And if you move on to the next slide, and then moving closer to, well, here we are in Central America and the story of maize domestication, which I think is probably, certainly, I think the most fascinating domestication story of all, because you see here the example of teosinte, the wild origins of maize, which is an unpromising, very small. The grains aren't particularly big. If you were to bite on teosinte, you'll probably break your teeth. But somehow, over a very significant period of time. We're talking about thousands of years and over a very large geographical area as well. So not just central Mexico, but the wild genetics started there. But thousands of years of humans interacting and selecting and exchanging to go from this unpromising weedy plant to the plump maize that is now one of the most important plants in the world because of the amount of nutrition that it provides. So a completely fascinating story from our deep history and how ingenious our ancestors were in interacting with these plants to provide the most important crops of all. So if you move on to the next slide. So I wanted to then look at the importance of diversity and trying to answer the question, is it really becoming endangered? If you move on to the next slide. And we have a character, a very important scientist, Nikolai Vavilov, who was a Russian botanist who traveled across 5 continents in search of plant diversity. So in the 1920s and 1930s, Vavilov was not only obsessed himself with the centers of origin, he actually coined the term centers of origin. So he wanted to map where the world's agricultural crops originated from, but also what happens when they move into different areas. So if one farmer moves and takes the seeds with him or her to a different environment, and they adapt to different conditions or they adapt to different human preferences, it creates diversity. So wheat, rice, maize, all of the crops that I've mentioned before started to really diversify. And Vavilov wanted to collect that diversity because when he was working as far back as the 1920s, he was concerned that that diversity was disappearing. If you move on to the next slide. Vavilov, it's an unfortunate and tragic story because he did incredibly, he did very important work in crop genetics, and the term genetics was actually coined at the beginning of the 20th century. So Vavilov was actually unraveling, unpicking why is it that there is diversity? What does diversity mean at a genetic level? He fell out of favor with Stalin in the Soviet period of the late 1930s because there was a Marxist form of science that had different explanations. And unfortunately, although Vavilov wanted to save this diversity to improve crops around the world, to ensure there was enough food for the future of humanity, he ended up in prison and died of the very thing that he was trying to prevent, which was hunger. So he starved to death in a Soviet prison. Next slide, please. So I followed in Vavilov's footsteps, and I wanted to go to the places, the parts of the world where he had traveled to see what type of diversity still exists. So I ended up in Eastern Turkey, for example, close to the origins of wheat. And I ended up meeting farmers who were still growing some of the original types of wheat that were domesticated 10,000 years ago, although it's now very, very hard to find. And they were saving it because it was important to their identity. They saw it as an inheritance from their ancestors. They loved the flavor and the look of these different types of wheat as well. So many of us think that wheat is wheat or maize is maize, rice is rice, but this huge diversity matters at a cultural level as well. And I'll get on to in a moment. They also have significant differences when it comes to nutrition and also disease resistance for farmers. Next slide, please. So one of the reasons why we start to see diversity disappearing is that we are able to start to take control of that process of diversity. So we are able to using science to select particular characteristics from a plant and actually turn that into something that many farmers around the world can grow. And this is exactly what happened or starts to accelerate in the 1950s and 60s. This is another scientist, Norman Borlaug, who was one of the chief architects of the Green Revolution. What Borlaug did was to take some of the diversity that existed in different parts of the world and to create a type of wheat. And he started his work in Mexico. His mission was to try and improve the livelihoods of poor farmers in Mexico, to give them something that was higher yielding, so it produced more grain and had disease resistance. So he took types of wheat from Mexico and the US, but also from Japan as well, a particular type of so-called dwarf wheat, which was quite short on the ground. What that meant is that could take up a lot of chemicals that had been developed during the 20th century. So chemical fertilizers, for example, pesticides. These crops that Norman Borlaug was developing produced a lot of grain, but they needed huge amounts of fossil fuel generated fertilizers as well. But they were so successful at the time that they quickly spread around the world, and they replaced a lot of the diversity that existed before. They also needed huge amounts of water for irrigation to produce the high yields that Borlaug had created these wheats for. So at the time, they produced a lot of food, but they came at a very big environmental cost. And this is why also that, next slide, please, another scientist called Jack Harlan, as early as the 1970s, was worried about what the world was losing in terms of diversity, that we were losing different types of wheat and maize and rice, but also humans have interacted and used for food around 6,000 different plants. Huge amounts of diversity. But what we see during this period of the 1950s, 60s, 1970s with these breeding programs is the focus in on the commodity crops of today. So the wheat, the rice, the maize. And so instead of depending on 6,000 different types of plant, the world now mostly depends on just a handful. And in fact, four crops provide the world with more than 50% of its calories, including wheat, rice, and maize, and a very, very narrow genetic selection of those crops. And just to illustrate what I mean by diversity, and this is the kind of diversity that Jack Harlan was interested in. He was convinced that if we lost that diversity, we create a fragile agricultural system around the world. And so he said that these resources, in other words, crop diversity, stand between us and catastrophic starvation on a scale we cannot imagine. And in fact, in the 1970s, there was a massive failure in the US Corn Belt because the maize, the corn had become so genetically uniform. If a disease hit one field, it could knock out all of the fields because there was no resistance. All of the genetic uniformity meant that the crop had become vulnerable to a type of disease. Next slide, please. So in terms of how much diversity is out there, it's very hard to measure, but a more recent generation of botanists listened to the words that Jack Harlan had said in the 1970s about the risk of catastrophic failure. And one of them, a scientist called Kari Fowler, was inspired to lobby governments in Europe, particularly Norway, to dig a tunnel into the Arctic Circle so that they could create a backup vault for all of the world's crop diversity and save the seeds in one place. And this is the Svalbard seed vault that we can see here. So when we say how much diversity is out there, hard to measure, but in Svalbard, there are more than 200,000 samples of wheat that have been collected from different parts of the world. There are more than, almost 40,000 samples of maize that have been collected from different parts of the world. And you can see the amount of diversity that exists around the world. But most of this now only exists in seed banks and in the seed vault, because this diversity has disappeared and continues to disappear from farmers' fields. Next slide, please. And this is not a world that many of us encounter, the idea of seed diversity, but it's extremely important and highly political. So here we have an image of the UN's Food and Agricultural Organization, a meeting that took place last November, where different nations around the world were talking about how do we share and how should we use the crop diversity that currently exists, because most of the agricultural diversity exists in the global south, and also places where there is still subsistence farmers, but they are sometimes the people who are, or the nations that are not necessarily the beneficiaries of a lot of the research that goes into producing future crops. So actually parts of the world that have saved diversity for a long time were not the beneficiaries of the scientific research that was based on the fact that these seeds had been saved generations after generations. And there were countries in that meeting, including Brazil, that were making the argument actually that it's important for countries not only to save this crop diversity, but to also have a say and have some control over how that biodiversity is used. And it's an ongoing process and highly political process. But what we now understand is it really matters, which is why I want to get onto the next slide, the why should we care. So and if you move on to the next slide again, please. So sorry, the slide before that actually. There is an important point to be made, and this is what I try and in my book Eating to Extinction, I traveled to many different countries, and I include around 40 different foods in the book. And I'm about to explain why there is a really important scientific case for food diversity to be saved. But it's really important to stress the idea that food is culture as well. It's part of our story. It's part of our identity wherever we are in the world. We often try and give a sense of who we are through food, through cooking, through recipes, from one recipe being passed on from one generation to the next, a seed being passed on from one farming generation to the next. This is a very important point in a world which is becoming increasingly uniform. A process of homogenization is not only resulting in more of us listening to the same kind of music, watching the same kind of films, wearing the same kind of clothes, reading the similar types of books, but also farming in the same way, eating the same kind of foods, eating similar diets. And as we know from the previous talk as well, this is coming at a high cost to planetary health and also human health as well. Diversity matters. It matters culturally. It matters in terms of identity. And I'll just explain now why it matters in terms of our future as well. So next slide, please. So if you think about something like a glass of beer. So, one of the world's most popular drinks. And there are countries around the world which have huge amounts of diversity. This is an image from probably one of the smallest bars in the world. It's in Belgium. It is a place where they sell a type of beer called lambic. Lambic is made with wild bacteria, so it's a spontaneously fermented which means it sometimes has unpredictable, crazy flavors, which the brewers then blend together. So there's the skill of making the beer, and then there's also the skill of blending it. But it's also a story of farming as well, because this is a type of beer that comes from very particular types of grain. What we see in the second half of the 20th century is the disappearance of this type of diversity of beers. So we've seen a resurgence in craft brewing, but certainly in the second half of the 20th century, what we saw was around the world, a small number of very powerful companies buying up lots of small breweries. And we see the disappearance of small breweries and the rise of one of the, of global giants. So AB InBev, for example, 1 in 4 beers drunk around the world is the product of that big global brewing operation. So we see consolidation globally, greater control over what used to be very local and very traditional to becoming a global commodity, even though sometimes the brands might look as if they're local and traditional, they're actually probably owned by a very big parent company. Next slide, please. But diversity is important for our future, and I would argue for our future both in terms of planet and people as well. Next slide, please. So we are seeing increasingly around the world a huge amount of fragility in the global food system. The global food system is ingenious, incredibly fast moving, which is why sometimes we see problems in the supply chain, and we saw some very big problems in supply chains during COVID. But what we do not see is the process that's actually unfolding around the world in farmers' fields. So why, for example, does that wheat in Turkey matter that I mentioned earlier? Why is it that the seeds that Vavilov collected, or the seeds that are in Svalbard, why do they matter? Well, it matters because the modern wheats that were developed during the Green Revolution were so successful that they spread around the world, and they worked. They worked for a period of time, as long as the fossil fuel fertilizers were there and the irrigation was there, and they worked as long as the environmental conditions were in their favor. That is now changing. What we are now seeing, and this is happening more rapidly because of climate change, we are seeing the rise of some extremely problematic crop diseases. So there's some images of wheat that is now suffering from, there's wheat blast, there's wheat blight. I talk about this very sneaky type of fungal disease in the book, Fusarium head blight, which can trick its way into the wheat and force it to commit suicide so it can take all the nutrients. This is spreading around farmers' fields, costing farmers billions and billions of dollars. And it's a problem because of the amount of uniformity now in the global wheat crop. So if you move on to the next slide, these are scientists based in Cambridge in the UK who are now trying to solve that problem by bringing diversity back into the global food system and the wheat crop. So what they're doing is they are taking the types of wheat that I saw in Turkey, and they're using it in breeding programs to almost take wheat back through its evolutionary history to bring more diversity into the crop, more diverse genetics, so that it actually has greater resistance to those diseases, that they can handle longer periods of drought or even flooding. So as I mentioned, the modern wheats developed in the 1950s and 60s were like the Formula One wheats of the world. If everything was in their favor, they would produce huge amounts of grain. We're not living in that world anymore. We live in an unpredictable world. So we need diversity. We need genetic diversity, which is exactly what Nikolai Vavilov saw in the 1920s and what the other botanists saw in the 1920s and what other botanists saw in the 1970s. Next slide, please. We are also losing diversity that we don't even fully understand. So I'm just going to take you in this next story to Oaxaca in Mexico. And this is a remote village area, home of the Mixe people. This is farmers who have been in this region of southern Mexico for thousands of years. Next slide, please. Botanists arrived and they saw this very strange looking maize in not very good soils. It was 16 feet tall, very tall maize. And not only did it have roots under the ground, but it also had aerial roots sticking out above. And from these aerial roots, as you can see, they were dripping this very strange looking mucus. And this was a mystery to the scientists who made it to this remote village in the 1980s. Move on to the next slide, please. And they couldn't figure out why was the plant producing these roots and what was in this mucus. And it was only in the last 6 or 7 years that they had the analytical techniques to show that the plant is feeding bacteria that live in the mucus sugars, and in return the microbes are fixing nitrogen from the air and fertilizing the plant. Now this is a interaction between a plant and a bacterial community that we find in leguminous plants like beans, for example, where you have a microbial population that can fix nitrogen and feed the plant. We do not see this in cereal crops like maize and wheat and barley. This was an incredible observation of the scientists, but the indigenous people who had been growing this maize for generations, they obviously had been interacting with this plant for hundreds, if not thousands of years. I mentioned the problems of the modern wheats and the modern crops of being so dependent on fossil fuels now. It could be that there were more plants like this that humans interacted with, but they are now disappearing. And this was one of the last villages to be growing this type of maize. So it's an incredible example of a type of food diversity that, again, we've only recently understood how it actually works at a scientific level. Next slide, please. We are also saving diversity that could mean that the possible devastation of not only one of the most wonderful and one of my favorite drinks, which is coffee, but also the livelihoods of farmers. And obviously I can't overstate how important this story is to Brazil. But what is important to understand about the journey of coffee from its center of origin or diversity, which is Ethiopia, so East Africa, where you have wild forests of coffee, a small number of plants traveled around the world to create the coffee belt around the tropics. And so even with the global coffee crop, it's quite genetically narrow, which is why we've had outbreaks of the rust disease, the fungal disease, which has really devastated a lot of coffee farming in parts of South America. For example, Ecuador was hit very hard by this. India used to be a very big coffee grower, but actually its coffee crop was wiped out in the 19th century by this disease, which is why India became more of a tea grower. And the problem is now that with the modeling about what the impact of climate change will be on global coffee production over the next, well, let's say over the next half century, it's a very serious problem, because we depend on two types of coffee, as you probably know, the Arabica and the Robusta. Arabica is quite a fragile plant and doesn't like variations in temperature, and it certainly doesn't like temperatures to be increasing, which is why it's a higher elevation coffee. Robusta also suffers when there is difference in precipitation. So, you know, if there is a change in rainfall. But it turns out Arabica and Robusta are only two of what are thought to be around 120 species of coffee. Next slide, please. One of the ones that actually almost, well, it did pretty much go extinct from commercial cultivation was Coffea stenophila in Sierra Leone. And this was a coffee that had been recorded in the 19th century by travelers, and it's as delicious as Arabica, and it's also quite disease resistant, more drought tolerant than Arabica as well. And scientists from the Royal Botanic Gardens in Kew traveled to Sierra Leone to see if they could find some of the surviving trees. They found one tree near a village, but finding one tree is like finding one panda bear. You need to reproduce and to have more seeds, you need to have two. So they actually went to some of the remote mountain areas and luckily found another. So Coffea stenophila is now coming back into production, and possibly in the decades to come will become as important as Arabica and Robusta, because it has that higher level of tolerance to climate change. So yeah, diversity does matter. There are major projects underway where scientists are working to save them. Next slide, please. During the COP process, so during recent COPs, the UN process, where we are trying to not only deal with climate change, carbon emissions, but also there are the COPs that are dedicated to biodiversity. At COP 15, which was held in December 23, there was the Global Biodiversity Framework. And that is a commitment to save 30% of the world's biodiversity by 2030. It's really important to note that this isn't just about saving a lot of what we might think of exotic wild biodiversity of birds and the flora and fauna of tropical forests, for example. The Global Biodiversity Framework also recognizes the importance of the plant diversity humans have developed through farming over 10,000 years. So there is a global commitment to try and save as much agricultural diversity as possible, because we now know how important that will be to our future. Possibly the most famous story of the consequences of uniformity is in the global banana trade. So although there are 1,500, possibly 2,000 types of banana around the world, we've ended up with one globally traded banana, which is the Cavendish. And as I mentioned with wheat, as I mentioned with coffee, the diseases out there can really undermine global production, and it really is doing that with the Cavendish, because the Cavendish is the second globally traded banana. The first one, the Gros Michel, actually fell out of production in the 1950s because it was completely overwhelmed by a type of disease, a fungal disease called Panama disease, TR1. And we now have the Cavendish, but the problem is we now have another iteration, another a variant of Panama disease called TR4, and that has now arrived in the Americas. So we are now extremely globally. It's a worry now that we have a disease, and the banana is a clone, so it's not propagated from seed; it's propagated from its suckers. So each banana is exactly the same genetically as another. If that fungal disease hits one area of soil and infects a plant, it can spread rapidly. There is no inbuilt protection from the plant. It's lost its defense mechanism because of the way we've taken over complete control of the genetics of that plant. We need more diversity. And I've spoken to scientists who practice conventional breeding, and also scientists who are using gene editing and gene transfer as well. So some of the really latest techniques, that whatever course they're taking in terms of trying to find a solution to the problem of the Cavendish, they all agree that we need more diversity in the system. Just to finish off, Diversity also is an opportunity for communities and farmers. Next slide, please. I mentioned about the Neolithic package. It included the lentil. There is a wonderful story of the lentil as it travels around the world, including to a place in southern Germany. Next slide, please. This is a farmer. Next slide, please. Sorry. This is a farmer who lived in a community high in the mountains, and they historically depended on growing lentils with wheat, and it created a type of dish, but also a farming system as well. It went extinct in the 1960s. He believed that he'd lost flavors and also his part of his identity and a farming system as well. So he traveled all the way to Russia, to Vavilov's seed bank, and there he was reunited with this lost lentil. If you show the next slide, please. So you had this wonderful, joyous moment of the farmer being reunited with the seed, which he then took back. Next slide, please. And he created a business and inspired other farmers to look back through their peas and beans and what had been lost from their diets as well. This is very important because of the dependence we now have globally on meat production, which we know is problematic on so many levels. All diets around the world, actually, in terms of protein, we had greater dependence on legumes, peas and beans, and that's definitely something we should bring back into our diets for health reasons, and also something for soil health as well. So it's an inspirational story of the past that should be able to help shape the future as well. Could you move on a few more slides, please? Because I'll just speed up to get to the end. If you can go to the diversity in health. This is a musician who sold his music rights to become a wheat farmer. Another story that is showing how mainstream diversity is becoming, because he started to create a type of bread in the high street that is now telling the story of diversity. But if you move on, please, a couple of slides, and again, and again, and again, sorry, and one more. Okay, so I'm conscious of the time, so I just want to finish on a really important subject. Why should we care? I hopefully have explained at an agricultural level why we should care. I also argue that we should care for cultural reasons as well. For people to come on board and engage with this, I also think we need to tell them a reason that will motivate them, and possibly the most selfish of all reasons, which is their own health. Next slide, please. So we know with the emerging science of the gut microbiome, for example, we know so much more, but there is huge amounts we don't fully understand. But what much of the science is telling us is that the gut microbiome does exert a significant influence on our physical and mental health. We also know from many studies, and there's a very well-established twin study in the UK showing that the greater the diversity of our gut microbiome, the more beneficial that is to our health, and the more diverse our diets, the more beneficial that is to our gut microbiome. We also know that when, for example, archaeologists have excavated intact bog bodies, so extraordinary bodies that are intact to the point where you can actually see what's inside their stomachs, huge amounts of plant diversity. So this is one that was, it's a body that's more than 2,500 years old, excavated in the 1950s, and more than 40 different plant seeds inside that one individual's stomach. And I think wherever you look in the world, there is a huge amount of diversity in traditional diets. And as I mentioned, we depend now on a very, very narrow genetic base in modern diets. Next slide, please. There are also studies now that are showing the actual microbial community around the roots of some of the so-called landrace crops, so the more traditional varieties. They actually are, not only does this microbial community around the root system mean that it influences how they grow, and how they take up nutrients from the plant, but also this could have a knock-on effect on the nutrient density of some of the plants as well. So, a modern wheat, we are starting to kind of learn now, the science is starting to appear and emerge that, there are reasons why there might be a completely different nutrient profile from a modern wheat to a more traditional wheat and other plants with deeper, more complex root systems. And next slide, please. The final slide. So the Rockefeller Foundation was one of the founders of the Green Revolution in the 1960s. And more recently, it's the Rockefeller Foundation that is funding this fascinating attempt to try and unlock the complexity of chemical compounds and I guess the so-called dark matter of food. So a lot of the macronutrients and many of them micronutrients we've been able to measure and we've been able to track, but they are just a fraction of what we now can measure. And so this now international effort with 7 labs around the world is trying to unravel what are the chemical compounds in a carrot. How does one type of maize grown in one farming system differ to another in terms of its nutrients? And again, the phytochemicals and all these other compounds that we now know are beneficial to our health. And the more information that we will be receiving in the years to come, because I think this is inevitable. We carry around computers in our pockets now. There is the rise of personalized nutrition. My argument is that I think that the more aware that we become of what's in our food and importantly what's not in our food, that diversity will become something more familiar as something that has been important in our evolutionary history that has been lost in many parts of the food system that we all need to regain for all of the reasons that I've mentioned. Final slide, please. And I think I've been surprised at how engaged people have been, because my book now has been published in now more than 10 countries. I think people, when they come across this untold story of the importance of agricultural diversity, it makes a lot of sense to them. And there's a completely different way of seeing the world and seeing our food. Thank you.
Thank you so much, Professor Dan. Wonderful presentation. Very, we are very sensitive with your words, especially because you introduced us a new concept, because here we are more involved in healthy area, to clinician, to research, something like that. And I am a pediatrician, for example, and we are suffering of nutritional problems, undernutrition with hungry, overnutrition with obesity, micronutrient deficiency, and these conditions are associated with is associated with chronic diseases and injury related to neurodevelopment. So when you talk about the diversity in agriculture, was like a new key that we needed to introduce in our knowledge, in our teaching, in our research. Thank you so much.
Thank you very much. Thank you.
And we are open to questions, please. It's a very important moment. I don't know if we have some here. Cristiane, Matté.
Yeah, while people are starting to warm up, I would have a question.
Briefing.
Thank you very much for your enlightening presentation. It's really, really, really nice. I already know your podcast, your program, and it's really nice to have you here. Thank you very much for spending time with us. So here, to have an idea, we are 4 professors in different countries, me and Fabíola from Brazil, Erika from Mexico, and Paola from Chile. And we have more than 100 students enrolled in this course. from 7 countries in Latin America. So we are really happy to have you here today. And we are thinking, our society works with development and environmental factors, including diet, that affects development. So do you think that this low diversity that we're facing today maybe could be related to allergies impacting the health of the people, because we see less and less diversity available on the markets, a lot of ultra processed food, and a lot of sugar, a lot of fat, and in fact we have a rise of obesity, a rise of a lot of disease, includes allergic disease, that maybe they are related to this small variety, small diversity we have in our plates. Do you think that this could be related?
I think, yeah, I do. And I think when this is why I think understanding the history, understanding the story of where our food comes from, and the idea, and in my work, and particularly in the book as well, I mean, I tell the story of, different types of timeline, but 2 million years of bipedal humans, and then 300,000 years of Homo sapiens. The vast majority of that time as hunter-gatherers. It's only in the last 10,000 years of that, which is a blip in, really, if you look at the long sweep of evolutionary history, that we became farmers. And then in the last century, we actually had the technological and scientific ability to completely dominate nature and the way we produce food, which is why our diets have changed. And you could argue our diets have changed more in the last 50, 60 years than they have done in the last 9,000 years. And I think, so in a sense, we are all guinea pigs of a system that is novel. Not only novel, and I've told the story of the way in which the genetics of our food has changed very quickly. So the idea that Borlaug could create these types of wheat that are designed for yield, so they're designed for abundance. They were not designed for flavor or nutrition. That was never part of the project. They were designed for quantity, and in fact produced for, they created so much in terms of quantity that we were then able to do more novel things with them. So we could use the excess of grain to create huge amounts of meat, which distorts our diet. We could also use corn, maize, and invent processes to create foods that are, my parent or my grandparents would not recognize. So I think understanding how quickly things have changed and how disconnected the modern food system is to the evolutionary story, how can it not have an impact on our long-term health? And obviously the global population has increased. In some parts of the world, lifetimes have increased, but actually they're starting to go the other way now. But we have a whole medical environment that we now exist in, which our your previous ancestors didn't have. But it's such a novel thing that we can't even, well, we are starting to see the long-term impact of this massive shift because of the micronutrient deficiencies or the obesity. We're starting to see it unfold at a frightening global level, which is why here I am talking to you from the UK, where type 2 diabetes is something increasing at a pace that is going to cripple, is going to really harm the national health system. And we are dealing with the symptoms. So we are medicating our way out of it, which is what came up in the last conversation. Whereas we need to get back to understanding the journey we as a species have been on in terms of food and farming.
Perfect. Thank you very much.
More questions? I just have one. Professor Dan, I think that you talked for us that the food is culture, and now you repeat that the grandmother teaching something for the daughter or the son about cooking, about the culture, about the food. And after that, we are following this way. I think we lost something in this way, the culture to learn with the family. And do you think this is important? What can I do to improve the communication about food culture inside the family? For example, my grandmother cooked more than my mother, and my mother cooked more than me. Understand how we can continue our evaluation in this way to improve our health, of course.
I think for me, and certainly how I see my role, is we need to know the story. So I think in many parts of the world, this is not a conversation. This is not a familiar subject. Many people do not think about farming, agriculture, where their food comes from, the stories I've told about how much change has happened so recently. So I think I would argue that whole story I told about, Vavilov, the domestication of maize, and all, this should be taught in every school. I think children should be growing up aware of how we as a species have survived, and we've managed to adapt, and our ancestors were ingenious to create the foods that we benefit from today. I think that can inspire people to really care about diversity. I also mentioned about the fact that I think we have the most selfish reasons of all to care about diversity, which the scientific evidence is growing now, that actually it does make a difference to our health, and that I think can motivate them. But I really do think, again, just to echo what had been said in the last talk about a lot of this needs to be top down. The food system now is so powerful, and the food companies are so powerful. It really is only now governments, or in fact cities, that can make a difference. And I know there has been some extremely progressive policies in Brazil, for example, where and I don't know the latest on this and the ups and downs, but I think there have been initiatives where a lot of school food had by law had to be sourced from smaller scale family farms. And I think public procurement is a really important thing. So what types of food city governments or national governments buy to feed children in schools, patients in hospitals, elderly people in care settings or whatever, that is a powerful economic trigger that can start to really make an impact on the wider food system and send out a message to farmers to bring back some of the diversity as well. But also think about the global subsidies that are now in the hundreds of billions of dollars. So public money is used to prop up the system we now have of monocultures and commodity crops that are processed into unhealthy foods. That exists and is sustained because of public subsidies, government subsidies. And I mentioned the Global Biodiversity Framework from COP15. At that event, they also said that we need to change agricultural subsidies around the world to try and improve the quality of food around the world. There are things that individuals can do, but with these very big global forces and the economics, it really is going to start to need structural reform. And it's only in the last couple of years we've started to hear that from people engaged and involved in policy, which is one reason why I think there is reason to be optimistic.
I am optimistic too, Professor. Margaret, you are with your hands, please.
Boa noite. O professor já falou quase o que eu ia falar sobre a questão do mercado, desse movimento do mercado, dessa produção de alimentos em série, pobres em questão sensorial, pobres em afetividade, sobre a questão de menos tempo na mesa que a gente passa, do tempo que a gente tem que estar correndo, as crianças na escola o dia todo, então se perdeu essa estar junto, comendo, coisas de família. quando você reduz o café da manhã a pote de cereal com leite, né? E não mais eu partir o pão, fritar um ovo, comer um queijo, um cafezinho feito na hora. Era mais ou menos isso mesmo, professora.
I will translate for you, Professor Dan. Margarete, where are you from? Você está onde, Margarete?
Fortaleza, no Ceará.
She is in Fortaleza, Ceará, the north of Brazil. The beach, good food, excellent food, Fortaleza. Professor Dan, Margaret talk about that she agree with you, and we lost, not everybody, but a part of us, lost the time to share food, the cultural talk, and for example, the breakfast is important. It's important meal, not just because we eat cereal with milk, but because we eat our food with friends, with family. So she totally agree with you, and it's interesting because she's in a part of Brazil that is totally different of, for example, where Cristiane is, because the Cristiane is in South Porto Alegre, and it's totally different. So I agree totally with you, and everybody here, not everybody, but a lot of people like coffee a lot too.
Yeah, completely also understand that economies, society has changed beyond recognition, and the way our parents and our grandparents lived and their relationship with food, some of it actually was itself problematic as well. I mean, many people in early generations didn't have enough food for various reasons as well. But we do live now in a world in which we are more fragmented, more disconnected, and more I guess separate in a sense. And I guess the economic pressures on people around the world means that our relationship with food is increasingly being tested. But actually, there are many of the world's problems. In my book, I'm saying, and I tell the story of 40 foods, and I try and tell some of these big trends through the 40 foods. What I'm not saying is that the 40 endangered foods that I talk about will feed the world, but actually we will have a better world and a better food system if they were no longer endangered. And I think we would have a better quality of life. Our societies will be better if the conditions existed where we could have a better relationship with food. And you could argue that is more time spent as family around tables, less messaging and less pressure from companies marketing to us and our children. And also, I think the idea that farming has become an industrial commodity process, whereas actually farming in communities is a really important feature throughout. Just a few generations ago, farming would have been such an important thing, and it hasn't been sufficiently rewarded. It's been cheapened. Food has been cheapened in every single way in our lives, but also the costs are very high now of the impact of that food system. So even if it looks as if it's a short-term benefit to have cheaper and cheaper food, and with the current cost of living problems around the world accepted, our food has become cheaper and cheaper and cheaper, but the costs have become higher and higher and higher to the planet and to our health as well. And that this has to be something that politics has to deal with.
It's perfect. Paola, please.
Thank you, Fabíola. Thank you, Dan. It's been a wonderful talk. Really helps us as scientists and reproductive biology people, most of us, interested in the reproductive process, to look outside the box, right? Because we're all wondering how we can stop this cycle. The name of the course is life cycle, and how this developmental origins of health and disease in the life cycle, right? And then the problem most of us are interested in is how we stop this vicious cycle of mom being obese, giving risk to the baby, and then the next generation because in the ovaries of the baby, you know. So one of the things that has been less considered, as you say, is farming, but also the preparation of the food, you know, because I'm from Chile, yeah, South America. And then the people that work at home, moms, grandmas, they stay home. They don't develop professionally, most of them, for the last generations. Now it's very common that people are studying and going into work. And then as Fabíola says, the grandma was cooking, the mom was cooking, and now we know less about cooking. And the problem is we're not taught about cooking. So if you're not pushed into cooking in your family, the society doesn't speak about cooking. So you go and eat to a restaurant, you go for other people to prepare, but nobody's promoting. You want to learn new ways to prepare your food. This is healthier, this is better. You can combine the flavors in this way, you know. So I think that's a huge gap in the society that we're living in, that nobody's talking about making it popular to be good at cooking, you know. So we should, our kids should be good at cooking. We should make cooking like a party. Families should gather around, not to eat, but to prepare the food to eat, you know. So how do you see this in the different cultures? Because other cultures probably, I don't know, some probably Indians and people from India, people from that side of the world are more dedicated to cooking as part of a ritual, a family ritual. Where do you see that we're missing? Where did this gap begin? Starting from the African origins to where we are, to where all the roots were spread. Where did we miss this part of the story?
And I've tried to, I guess, explain how some of that happened. And I think there are more structural things about global trade as well that unfold, and corporate power can't be ignored as well. But I think what we've ended up with is a situation in which we have completely this siloed approach to policy that should be really joined up. So, for example, health policy in any government, you know, health department should be linked to the Department of Agriculture, you know, in terms of what you grow and what you produce when it comes to food is going to have an influence on the health of the population. But these are completely, in most governments, they're completely separate, and there is no connection in terms of policy. And you could also argue that the business department is different to the health department and different to the agricultural department, and therefore, in terms of trade and trade barriers and tariffs and that kind of thing, you know, it's very, you know, easy for, you know, one bit of government to make it easy to import really cheap, unhealthy ingredients, only for the other department, the Department of Health, to actually pick up the bill and actually to have to resolve that in. And then, as you said, you know, the Department of Education should be linked to the Department of Health, and in which you really understand the importance of food education and school food when it comes to, you know, healthy children, healthy brains, you know, omega-3 fatty acids in school food diets, and behavior in classrooms as well. None of this is, mostly in most governments, it's not joined up, and this is, and it's ludicrous, because again, you know, the high financial cost, the high cost in terms of human unhappiness and lack of health and environmental problems as well, none of it is joined up in most parts of the world. And unfortunately, including in India as well, which is seeing soaring rates of obesity and diabetes, huge amounts of processed foods coming in because it's seen as an desirable, affluent, you know, type of food, you know, that you should be eating instead of traditional diets. It's not like it's a huge country, it's a huge population, not always the case, but in urban areas, food is now, you know, really problematic in terms of health and obesity and diet. So I think it's this lack of connectedness now, because food became fuel for, you know, societies chasing economic development without understanding. You know, there are periods in our history where we didn't have the knowledge, the evidence, which is exactly how governments then started to make a difference to smoking and tobacco, for example. But we now know the cost of bad diets on populations, and hopefully maybe we will see more and more aggressive government action, because even if they just think about the budgetary impact of these bad diets. So but I think we need cleverer governments, and cleverer, and maybe even at a city level. Cities are where some of the most progressive experimental policies around food have taken place, because things are so slow to happen at a global and national level. So I think at a city level, it can really, you can start to see in London, and I'm sure in some cities in Brazil, there are some really progressive policies. Across Europe, you're seeing it as well, because there is an awareness that you can, at a city level, start to make a difference.
Thank you. Bárbara, we are for hands up, please.
Eu só queria fazer um comentário no que ele falou. Eu sou nutricionista de uma escola aqui em São Paulo, e já fazem 3 anos, e eu vejo como as crianças, elas não conhecem os alimentos, assim. Eu tenho muito problema, porque crianças, assim, de 10 anos, às vezes a gente tá ali fazendo uma educação nutricional com eles, alguma atividade, e eles não conhecem, assim, comidas muito simples, como mandioca, mandioquinha. Então, assim, eles realmente não estão acostumados. Você vê o lanche que eles trazem de casa, é sempre um bolinho pronto, aquele suco industrializado. O que for mais prático, assim, você vê que eles acabam trazendo, e um pouco de conhecimento, que você conversa com eles, você vê como eles experimentam e eles acabam gostando, né? E eles acabam, tem crianças que eu tô já assim 3 anos com eles, e você vê a evolução, assim, sabe? De quando você faz alguma coisa que você introduz esse alimento e eles acabam gostando e comendo. Então você vê que é uma coisa que vem de casa.
Which part of the country are you from Brazil, but which part?
Eu sou de São Paulo.
São Paulo. Professor Dan, she's a dietitian. She have worked in a school with children, and it's a very interesting comment, because she told for us that a lot of kids don't know foods, general food, banana, manioca, foods in general, and they started to know at school, start to know, or the school introduced the foods, natural foods, not just in the diet, but in the education for young children. So the school is very important in this way.
We have many examples, well, not many, but we have some really important examples in the UK, where charities have got access to funding to do really fun, informative work with the youngest children in the education system. So they make time where they can use food to teach maths, they can use food to teach history, and then they have in one school that I know particularly well, and it's often down to individuals. So the head teacher is extremely passionate, to the point where this school has greenhouses where the children get to handle seeds, and they watch food grow. And I think if you give children the opportunity, they will become fascinated, and they will then become engaged in food, and they will care about food, and they will even go home and tell their parents things about food that their parents don't understand as well. Where it has been done in an imaginative way, children do become really engaged, and we have some award-winning programs in the UK where there is evidence of children really starting to become switched on and excited about food at a really young age. And that's where you have to really get the kids involved at a very young age, which will then hopefully, well, it will, that, because there's no point trying to teach obviously you can try and teach a 15-year-old. If you can get children at a very young age of, school entrance 4, 5, 6, then I think that can shape their future relationship with food in a really significant way.
It's perfect. Aline, please.
Oi, eu também sou nutricionista, assim como a Bárbara, e como ela falou um pouco sobre a questão da alimentação na escola, né, vou falar a minha experiência de trabalhei com alimentação escolar, mas em escolas municipais, municípios pequenos, sou do Rio Grande do Sul, e é bem diferente, assim, da realidade dela. Então, as crianças conheciam, então, um município de 5.000 habitantes no norte do estado, do Rio Grande do Sul, né? As crianças conheciam a alimentação, mas a gente tinha bastante dificuldade, era através da política pública, né, no PNAE, a gente tinha bastante dificuldade de conseguir alimentos da agricultura familiar. Então, aí entra em outro ponto, que tem uma obrigatoriedade, que 30% no mínimo do recurso tem que ser usado com esses alimentos. E aí tinha dificuldade por não ter produtor ou por não atenderem as demandas. Então, uma realidade um pouquinho diferente, mas que também vai bater nessa mesma questão dos alimentos aí.
Thank you, Aline. Aline, Professor, is a dietitian too, but she have worked in small cities in south of the country of Brazil, Rio Grande do Sul, small cities. And she is talking about that in small cities, the children knows better the name of the foods because the life is good in these places, but there is a problem because we have a public policy here in Brazil. It's a very important public policy about the food in school, and it's a rule that 30% of the food at school. It's important to bring it to agriculture, familiar agriculture. But besides a rule, there is no adequate number of the familiar agriculture around this place. So it's very interesting because the place it's good for this. We have laws for this, but it's not easy to identify the groups that are involved in this. Very nice.
Yeah,
very nice.
Which shows how interconnected. Yeah, shows how complicated and interconnected. So, that whole supply chain. You need to have the farmers, the small-scale farmers, with the diversity of crops, who are being sufficiently rewarded, so they stay farming. Which is why I think public procurement, so city-based budgets, if used well, can actually keep those farmers farming. But it's a global problem. We are the number of farmers around the world is shrinking, and the farmers that are still there are getting older. So, and what could be more important than the future of the world's food? And somebody has to be growing that food. So it's a crisis of the future. We need farmers, future generations of farmers, and with the use of technology, and I think with more fair, just financial rewards, we can have young people who are going into farming, who want to be still on the land and having a high quality of life themselves. And again, it's just we are not placing enough value on food. That ultimately is the problem.
Yes, I believe you. So thank you so much, Professor Jan. It was a honor for us to receive you, to have you with us during this time. I hope to see you in person in England, in Brazil, and or other Latin American country. Your words, I wish that everybody read your book, should read your book, follow your Twitter and Instagram and BBC documentaries. And the last words, it's for you, Professor Dan. Thank you. The last words for you, and thank you so much again.
Well, thank you very much for inviting me to talk about these ideas. I think whatever your area of specialism, I think this is relevant. How can it not be? I think this is the biggest story in the world, in our lives. You know, the how did we get to where we are today, and the food that we have today. So I think it underpins everything. And I hope it's been helpful, and it's made you think about your own work in a different way. It's given you a new lens to work with in understanding our relationship with biodiversity, with the world, with plants, with animals. It's a fascinating story. So I hope you have time to learn more, and it can be a powerful tool for you in the way you think about your own work. And best of luck, because it's very important, the work that you are doing and the ideas you're shaping. So again, thank you very much for having me.
Thank you so much. Have a very nice day, very nice week. Everybody, see you next week. Thank you, Professor Dan.
Thank you.
See you.
Bye.
Bye-bye.
Thank you, everyone. Hasta la próxima semana. No olvídense del trabajo